Welcome to the 49th Annual Holiday Lectures at Rockefeller University. I'm Dr. Bonnie Kaiser, and I direct the Science Outreach Program here. Professor Sandy Simon will talk about life through a lens, what you can learn about biology simply by looking. I will go more into that after I tell you a little bit about the Rockefeller University. Since the university was founded in 1901 as the first biomedical research institute in the United States, Rockefeller scientists have made significant achievements in medicine and science. One of them was by Dr. Porter, who in the 1970s published this book on fine structure of cells and tissues. This is like learning about life by doing anatomy on cadavers, which is very, very important. Sandy will take us into a world that is just amazing. We will look at life as it is actually happening. A total of 23 scientists associated with the Rockefeller University have been honored with the Nobel Prize since the university's founding over 100 years ago. 4 of them in the last 9 years, including President Paul Nurse in 2001, Roderick McKinnon in 2003, Gunther Blobel, and Paul Greengard. The Rockefeller University Holiday Lectures in Science were established in 1959 by Alfred E. Mirsky, a biochemist and Rockefeller University librarian. Dr. Mirsky modeled these lectures on a popular series of science lectures for children, pioneered in London in 1826 by Michael Faraday, known as one of the greatest experimenters in the history of science. Now we move on to another great experimenter in the history and who's making history in science. Sandy Simon came to Rockefeller University in 1984 as a postdoctoral researcher working to understand how proteins are transported within a cell. His postdoctoral research was in the lab of the world-renowned biologist Gunther Blobel. Dr. Simon was promoted twice to research associate in 1986 and assistant professor in 1989, and established his own lab, the Laboratory of Cellular Biophysics at Rockefeller in 1992, which is when we started the outreach program. I'm very proud of the fact that Sandy was also an Annenberg professor, which meant that he was very much involved in science outreach, not only taking high school students into his laboratory, but teachers as well. Dr. Simon's bachelor's degree is in neurobiology from Princeton University. He also has a Ph.D. in physiology and biophysics from New York University Medical Center. For 3 decades, it's been a long time. For 3 decades, Dr. Simon's research has used imaging to examine individual events in biology, how single proteins cross membranes, how single packets of messenger molecules are released from cells, how single viruses assemble, and how single tumor cells spread. His work has led not only to new imaging techniques and technologies, but to new biological insights that could never have been made by observing only average behaviors. As an educator, Dr. Simon has helped design high school science classes, including portions of a curriculum in use in several New York City schools, and it was pioneered at the laboratory schools in Chelsea, where the outreach program is very proud to have 9 present and former outreach teachers. This curriculum is designed to teach students to approach scientific problems as scientists approach them in the laboratory. And now, Sandy Simon. Thank you very much.
Thank you for coming. During the lecture today, there are a number of different points that we're going to try to cover during the talk. If you wish, a number of different strands that are going to be going on in parallel. So first of all, I'm going to try to present to you sort of a philosophy of science, in which I'm going to cover a number of questions which at least bothered me when I was a student growing up and learning science, such as why are so many of the explanations that have been given to us by science teachers or by our textbooks wrong? Along with it, what can science actually accomplish? What are the kinds of questions that a scientist thinks about? Why should I pay attention to any of this if so many of the explanations have been proven to be wrong? For that matter, why should I pay attention to my science teachers if so many of the explanations have proven to be wrong? That's a question that's fair for the science teachers to ask as well. And along with it, given all of this, should I consider a career in science or not consider a career in science? Because in fact, science as we practice it in the lab is actually quite different from science as it's taught in the classroom. That's sort of one set of thread, this sort of philosophy of science. But a second sort of parallel thread is an approach to science. In particular, I'm going to tell you about an approach in which we emphasize studying single events in science. So very often when you study things, you do it by looking at the average. For example, what is the average age of, let's say, kids graduating high school, or what's the average weight of a typical middle-aged person? We don't look at the individuals involved. When we look at biology, very often we'll look at single cells, or we'll look at single organisms, and we'll ask, how do they behave? We'll look at the average behavior. So for example, when you do biochemistry, you'll accumulate many different proteins, and you'll study the average behavior of the protein. You won't study individual molecules and ask how they behave. And what I'm going to do today is give you some examples where we study either single molecules or single organelles, single parts of cells, or single cells themselves, or for that matter, even single individuals. And what we'll show you is that the kinds of answers you get, the kinds of questions you might ask, are actually quite different than if you're looking at the average behavior of either those molecules or the cells or even the individuals. Another thread that we'll be following today is the use of imaging. And if you haven't been following it, imaging has started making a tremendous impact on science today. So for example, 2 weeks ago when the Nobel Prize in Chemistry was given in Sweden, it was given to 3 individuals for identifying how to use one particular protein from a jellyfish to be able to label proteins in such a way you can make them fluorescent and follow them move around. Imaging is currently having an impact on all areas of biomedical research, from the level of looking at the activities of the single molecules all the way up to development of organelles and important clinical issues as well. So I'll go through some of the reasons why imaging is having such an impact. Along the way, I'm going to illustrate these points by using a number of different research areas. So for example, how do cells secrete? How do cells grow? How do cells respond to various signals in their environment? How are viruses made inside of cells? And how do tumor cells migrate? So these will be the model systems that I'll be using to try to illustrate to you some of these other points along the way. The conclusions that I'd like you to take home with are the following. There are certain things that you can learn from studying single events that you cannot get from studying the average properties of a population, and that imaging is allowing questions to be addressed that even 5 or 10 years ago were thought to be completely inaccessible to science. And so the very final conclusion is that this is the most exciting time ever in history to be doing science. So first, a little bit of background on what is it that motivates a scientist. So one trait that distinguishes humans from other animals is that we try to describe our world. Some people describe it through visual arts, pictures, sculpture. Some people describe the world around them through stories, or through song, or through drama, plays and movies. Others describe the world around them through philosophy, through religion. Each one of these ways of describing the world around them, we judge by how aesthetically pleasing they are for us. Is it something that I feel comfortable with? Do I enjoy? They aren't judged by whether they're right or wrong. You can't say a particular song or a particular play is right or wrong. The question is, does it somehow resonate with me in a way that's aesthetically pleasing? Does it give us a sense of place in the world? A second trait of humans is the desire to plan. And that has a very, very strong evolutionary advantage. And to plan requires much more than being able to describe the world around us. It means being able to predict what's going on. So for example, if I'm running through the woods and I'm being chased by a tiger, to be able to plan, for example, now if I swing across the river on a rope, will the animal that's chasing me be able to follow? Or predicting when will the Nile overflow? So if you can predict exactly when the Nile is going to overflow, you can then predict when to plant your crops or when to harvest the crops. When will the seasons change? So science is an attempt to use our descriptions of the world to predict what's going to happen. And so what science allows us to do is to come up with models that make predictions that we can then test and thus allow us to plan. So art, philosophy, theater, all of those are judged by how aesthetically pleasing they are to us. Science, on the other hand, is not judged by the aesthetics of its models. Instead, what science is judged by, and science is not judged by whether or not it's the truth. Now, this is an extremely important point. There's no such thing as the truth in science. This is something we'll return to. Science is judged solely on the accuracy of its predictions. So, for example, Newton. Newton could predict the rate at which an apple would fall from the tree. Now, we know that Newton's ideas can be used to do things like predict the rates at which all sorts of objects fall or move. For example, we can use it to predict the trajectories of rockets going far off into space. So are Newton's ideas of science are his models of the truth? Well, we know now that if you actually go to very, very high speeds or very small sizes, a lot of the predictions of Newton fall apart. But the question for us as scientists is, does it make useful predictions? And so I would say that even though it's known to give the wrong answers under certain conditions, it makes many useful predictions, and therefore Newtonian mechanics is what I would call a successful model in science. Note, I'm not saying whether it's the truth or not the truth. We judge science based on the accuracy of its predictions, not whether it's the truth or not. So in general, we never refer to something as a truth. It's a useful model or not. And part of the reason being, and this is something I'll give you specific examples of, is that we never know whether there are assumptions there that we're not yet aware of, assumptions that may undercut everything we're doing. So as a result, scientists never really know if they have the final answer to a question. There can always be yet another assumption. For example, Newton did not realize that once you went up near speeds of light, something that was not accessible in his time, a lot of his laws would fall apart. Or if you went down to the level of small atomic particles, it would fall apart. Thus, in science, there's no such thing as really knowing the right answer. And I'm going to love to discuss this with some of the teachers afterwards. So as a result, it's very important to realize is that very often there may be many other models that can be equally successful at predicting our observations. So the critical thing for judging any scientific model is it successful at making predictions? So given this, how do we proceed in doing science? First, we make observations. Based on these observations, we make various models from which we try to make predictions. And advances are often made by coming up with new ways of making observations that test the predictions. So very often you'll hear about major leaps in science. In fact, Nobel Prizes are often given for people who've come up with a new way of making an observation, a new technique, a new instrument that allows us to test models that previously could not be tested. So an extremely important point, when you end up disproving a model, that's a sign of science at its best. It's a sign of science being healthy. And unfortunately, very often in the media, when someone's disproved a sort of favored model in science, the response you see is that, oh, science was wrong. Scientists have made a mistake. But in fact, it's inherent in the process that what we're always trying to do is disprove the models as a way of making our models more predictive and thus more successful. You can work your entire life only near the end to find that your cherished model, which has been the basis for all of your experiments, has been proven wrong. The important thing is that the net result is that science becomes more predictive and then becomes much more useful. So for us as scientists, the most important thing is to generate results that are reproducible. What you want to be sure is that if any other scientist does exactly the same experiment with the same methods, they'll come up with the same answer. So in other words, if you took an apple and dropped it, you would get the same results that Newton got in his experiments. What are the kinds of things we worry about? Well, there are a number of concerns. One of the critical ones is the result that we are getting a consequence of how we did the experiment, something we call an artifact. So an artifact is some kind of error or misrepresentation that's the result of something in our technique or our technology. What you want to do is at least be sure that your result is a robust one that anyone else can get and is not due to some artificial consequence of how you're doing your experiment. You want to be sure are the results a consequence of the biology and not how you're actually doing the analysis. In other words, you want to be sure you don't have a mistake in your computer code that you're doing, or you haven't made a mistake in how you've plotted the data, or you haven't made a mistake in how you've analyzed the statistics of your result. Once you've actually made it through the experiment, then subsequent concerns are there assumptions in the experiment that we were not aware of, assumptions that could have dire consequences for the interpretation of our results? And then finally, are the results consistent with our hypothesis, or are there other hypotheses that are equally consistent and make the same predictions? What I'll do is I'll give you some actual experiments from my own work. This particular issue of is an artifact the result of misrepresentation is something that has been sort of a personal demon of mine and for many scientists. And I really want to sort of drive home that point because this is the kinds of issues we deal with every day when we do science. And the model system I'm going to present to you for sort of how I've been sort of dealing with this problem, how do I know if my result is real or an artifact, is from work we've been doing on how cells secrete. So actually, how many people here have never seen a cartoon of a cell looking vaguely like this before? Just how many people have not seen some kind of illustration of a cell? Most of you have seen it. And you're familiar, most proteins are made out here in the cytosol. Proteins that are going to be secreted from the cell. Does anyone actually know where they're made? RER, which stands for the rough endoplasmic reticulum. So here's the endoplasmic reticulum here, and actually the reason why you called it the rough ER is that if you looked in Porter's book that Bonnie referred to, what you would see are little machines sitting on here called ribosomes, which are making proteins. So once they're made into the rough ER, where do they go to next if they're going to be secreted? Okay, I heard a few people saying the Golgi apparatus. From the Golgi apparatus, they then make it to the surface where they're secreted. Are the microphones up right now? I've got the light in my eye so I can't see. Okay. Could someone just describe to me, how does this process of secretion actually occur? Is there someone who can get near a microphone? There's one on each side over there.
The substances are contained in these small membrane-bound sacs, kind of like bubbles, called vesicles, and then they move toward the membrane and they merge with the cell membrane and it expels the contents.
Okay. That's an accurate description from any textbook you'd read today. What kinds of things is secretion used for? Well, why would a cell secrete? So getting rid of waste. So actually first, this was your answer, right? A membrane-bound packet fuses, stuff on the inside's released to the outside. So what is it used for? Can someone get near a microphone? Say what secretion is used for in the cell.
It's used for the secretion of certain chemicals and such as insulin, and which usually help the body to function.
Okay, so the cell releases enzymes to the outside to digest, or insulin, which is a signaling molecule. So for example, later on when we have lunch afterwards to help you digest some of the food, as the sugar enters your system, you'll secrete insulin to stimulate the uptake of sugar, so that hormones in the circulation. It turns out that between nerve cells in your muscles, so for me to walk, each nerve cell has to secrete transmitter to stimulate the muscle. And if you go through the textbooks, they're also used adding a new membrane to the surface. So as was described here, when they fuse, they deliver membrane. You had new receptors. All of those answers are consistent with everything that's in all the textbooks. And one of the things that I want to drive home today is that, in fact, we understand almost nothing of how things work inside of a cell. So leave aside secretion for a moment. If you just look at where the nucleus is, which is where the genetic material is, we have no idea of how things get into the nucleus or out of the nucleus. If you look at the mitochondria here, sort of the energy powerhouse of the cell, we know there are certain complexes there that are essential for metabolism, essential for generating energy, where part of the complex is encoded by DNA here in the mitochondria, part of it's encoded by separate DNA here. These two separate sets of DNA have to make proteins that have to get together and assemble into a complex. We have no idea how that communication occurs. We have no idea how it's regulated. If you look specifically at the issue of secretion, which is what we're going to focus on today, just the simple question of how you sort things out. We don't know how cargo gets sorted to different targets. We don't know how to separate cargo that's needed to go out at a steady rate. So, for example, cells that are growing, they're adding new membrane at a steady rate. But other things you want to regulate going out. For example, the young woman over there mentioned insulin. You don't want to be continuously secreting insulin. You want to release insulin only when you've eaten some food and the sugar is higher in your blood. Or another example, the hormones that regulate the flight or fight response. You don't want to be secreting those all the time. In other words, you don't want to be sitting down to watch a television show and suddenly sort of be secreting the flight or flight. I mean, actually, maybe it'd be good if you ran away from the TV, but you might not want to do it. Your parents may want you to do it. You want to be able to separate out the things that you're releasing on a regular basis from ones that you want to control being released, and we have no idea of how these things are being separated. We don't know if there's a link between the machinery that's sorting the cargo into these separate vesicles and then the machinery that then actually targets the vesicles to their destination. In other words, do you sort them with one set of machinery and other machinery then carries them to the left, right, top, or bottom? What determines where a vesicle is targeted? So almost every cell in our body is highly polarized. It's got an upper part or a lower part. It's not symmetrical. It's not the same all over. And proteins get targeted to very specific destinations. And this is an extremely important point, not just for understanding basic biology, but also for understanding a lot of diseases. It turns out that many diseases are the result of a specific mutation that affects the sorting and targeting of a protein. Gunter Blobel, who was mentioned before, initially identified many of the sorting and targeting signals for proteins. So Gunter's calling to remind me that I forgot to say something about his work. That isn't me. So it turns out certain diseases, like cystic fibrosis, the most common genetic disease amongst Caucasians, is the result of a single point mutation in a protein called the cystic fibrosis transmembrane regulator. It turns out if you study this protein, so David Gatsby, who I share a floor with, studies this protein, and with a single point mutation, this protein behaves just fine. It's normal. Everything you look about it is normal. The problem is this single point mutation affects how the protein gets sorted. So it's functioning right, it's just functioning in the wrong location of the cell. There are many such diseases where you'll have a single point mutation so that the protein you want to study is totally fine, it's going to the wrong place in the cell. We have no idea of how this happens. So the answers to many of these questions were known when I was a student, both in high school and college. In other words, I was taught, this is how it happens. The answers are not known. So one of the questions that might be useful to address is, why are we not as smart today? Now, specifically on the issue of how the vesicle fuses to release its contents. So when that membrane-bound packet comes up to the surface and releases its contents, how does that happen without causing any leakage? Not known. We know there are various proteins involved and various lipids. We have no idea what they're doing at the time. Is it mainly the proteins doing the work? And if so, how? Or is it the lipids? When it starts to fuse into release its contents, does it always then go all the way releasing everything? Or do you actually regulate it, allowing little sort of spits of release going on at a time? I'm sure there's a proper scientific term for it, but That's what we use in the lab. How is the process regulated? Again, we have no sense of how this is controlled. We know that insulin, for example, can regulate the fusion to the surface of vesicles carrying a sugar transporter, but we don't know how that regulation occurs. Is there a linkage between the process by which you then fuse things in, delivering new membrane to the surface, and the process by which you then internalize things? So again, when I was a student, both in high school, college, and graduate school, I was taught what the answers were, but in fact, the answers to these things are not known. So my first introduction to the secretory pathway came from studying the Golgi apparatus, which was mentioned before. And I initially learned about it in high school. I grew up in a small town south of here. Just as a show of hands, I know that this is a science lecture, not a geography lecture, but how many people here have heard of Brooklyn? Okay. I went to Madison High School in Brooklyn, and there, in my ninth grade, my first science class, Mr. F, I will call him Mr. F. You will understand why in a few moments. It's not the reason for what you think. Taught us, and he used to teach us everything as a way of preparing us for the Regents Exam, which I gather you still have to study in the public schools. And at the time, the way the Regents Exam was set up is that everything was in the form of a multiple choice question. So everything we had in class was a multiple choice question. So what are some of the functions of the Golgi apparatus? You have to say it louder. Modify proteins. How are they modified? Carbohydrates, sugars get added onto them. What else happens in the Golgi? Packaging of proteins. Okay, so we would get exams where the answers would appear like this. It's the shipping center for the cell, the site at which all molecules to be exported get packaged for their final shipment. Or, these are multiple choice, B, a song by Fish. Or, the sweet spot of the cell, where proteins are modified by the addition of different sugars. Or you'd have to answer A and B, but not C, or B and C, but not A, or all of the above, or none of the above. Okay, so I see that a number of you have taken the Regents Exam before. Well, actually, no, there's a very good reason why it doesn't say A and C. So what happened was, when I was starting high school and I had Mr. F in ninth grade, I was determined to show him I really cared about science, and I was going to give him what I considered a scientifically accurate answer to it. And what I decided to do, instead of just answering by the letters, I realized there were actually caveats to each one of these. There are problems with each one of these answers. For example, the sweet spot of the cell. So based on some experiments that were being done, there were reason to think that this is where carbohydrates were being added, sugars were being added, but you had to accept that there were certain assumptions in the experiment. It was done by either breaking the cell apart, and you had to assume that you were accurately separating out the parts and accurately separating out the section of the cell. If you accepted all of those assumptions, then okay, then maybe C would be a correct answer. And what I proceeded to do is, on the exam, I started writing 2 or 3 or 4 sentences about each one of the answers, explaining why they were potentially wrong or potentially right. In the case of the sweet spot of the cell, well, it says where proteins are modified by the addition of sugars, but maybe proteins are modified by the addition of sugars elsewhere. So it turns out that back then they actually thought the only place where sugars were added was in the Golgi, something that the teacher pointed out to me. In fact, we now know today, it took 20 years before someone finally looked to ask, does this happen elsewhere? It turns out that most of the proteins that are making these little pores in the nucleus actually have sugar molecules on them as well. They're not added in the Golgi. And I started going through each of these answers. So the only one that I felt really secure about actually was B, which is why I didn't say A and C, but not B. Wait. It's a little funny. The line is, I looked through the microscope and saw the Golgi Apparatus. It's from Fisher's first album. So that one I knew was accurate. C could potentially be right, but so I ended up writing these long answers, handed in my exam, feeling particularly proud of myself. And when the exam came back 2 days later, since I had not marked down any of the letters on any of them, I had gotten a consistent grade of a 0 on the exam. So I felt as if I should be able to rationalize my way through this. So when I took the exam and I stuck it to the bottom of my bag, figuring when it came time to the next exam, I would rectify this with the teacher and clarify what I was trying to do. And actually, I should be careful what I'm saying, in part because both of my kids are here. So if you ever get an exam where you get a straight 0, do not put it at the bottom of your bag. You show it to me. But for the rest of you, what I did is, then for the next exam, I went on, and I ran into the same problem. And so now I had 2 zeros in a row in science, and these are one of these things which you sort of can't keep from your parents. These things do eventually come back, and this ended up, what I viewed as a success in biology, and that I was really learning about it, ended up being problematic for my mother for a few reasons. Number one, A, she wanted me to go to college. F's in science didn't help scholarship. B, she always thought she was special in my life. She thought I only caused problems for her. But now I was causing problems for somebody else, my teacher. Or C, she was a teacher in my school. She was a teacher at Madison, which meant that every day she had to face her colleague, my science teacher, Mr. F, who knew that her son was getting 0s. Or A and C, or E. So fortunately, winter break came, and Mr. F gave us the option of trying to redeem ourselves by writing a science paper on anything that we wanted to. So this was right at the time when George Pilati, shown here, working here at the Rockefeller University, was in fact working out all of the details of the Golgi apparatus. So Pilati was known for a number of things, but his 2 key contributions to the Golgi was that he was the first one really to characterize it under the microscope, under the electron microscope. And second, he actually had separated out, he'd broken apart the different parts of a cell, and he isolated and purified it. This was a magnificent tour de force in science. He was part of the first project ever to both separate the different components of a cell, identify them, and to identify them under the electron microscope. So I thought this would be a great subject. I was going to write about the Golgi. I would finally show him what it was about. And I managed to get a library card to the New York Academy of Medicine library. Spent the whole winter break there and put together one of these tomes of science. Handed it in, and I don't know if you've ever had a project that you were like really, really proud of, that you thought, I handed it in, and I was convinced the next day Mr. F would come in and say, you know what, Sandy, I'm sorry. It's clear that I completely misread you. I thought you were trying to be difficult. But in fact, okay, maybe you were trying to be difficult, but it's clear that you really do care about science. Well, the next day came and he didn't have the exams, the papers, and the day after that, finally about 10 days later, he brought in the papers. They were all sitting on his desk, and he went around the room putting them down on everyone's desk, and he got to my desk and put it down, and there on the top of it, in big red letters, was an F-. Thus his name. So I have no idea what happened for the rest of that period. I don't know if you ever had an experience where somebody tells you something that's simply so shocking that, like, there's a disconnect between your world and everyone else. And I missed the rest of the period. The thing's over, and he came up to me and goes, listen, I'm really sorry. It's clear that you put a lot of work into this, but it would be a mistake to reward you just for doing a lot of work, because it's clear that you misunderstood everything you read. He said, because everyone knows that when it comes to the Golgi apparatus is just an artifact. It's a blob that's observed when you color the cell with too much stain. So it turns out that in 1952, when Mr. F had been going to college, they actually thought that the Golgi didn't really exist. There's a period of time of about 9 months, shortly after they had first identified the Golgi, where they actually thought it was just the result of having too much of this stain, too much dye being put into cells that was just globbing together, causing a distinctive little blob there. It wasn't real. So move ahead now about 15 years. I'm walking down this main marble walkway of the Rockefeller campus. At this time, I had George Pilati, who was standing on one side of me. The other side of me was Gunter Blobel. Gunter was one of George's star postdocs. By that time, Gunter had his own laboratory here at Rockefeller. By this time, George Pilati had already won the Nobel Prize for his work on the Golgi. And Gunter Blobel, a number of years later, actually would get the Nobel Prize for studying how proteins enter into the secretory pathway to then get processed by the Golgi. And it turned out my parents were clearing out their house, and my mother had found this paper I had written, which I had saved. She wanted to throw it out, but I had wanted to save it. And I told George the story of how I'd gotten this F- writing a paper about what was really his sort of magnum opus, his study of the Golgi and how it functions. George thought it was very cute. He actually asked to have the paper. He actually kept the paper with him in his office until he unfortunately passed away about 3-4 weeks ago at the age of 97, was it? And so as he was looking through the paper, Gunter sort of took me by the arm and said to me, "Sandy, in 1952, it was George Pilati who said that it was an artifact of how you prepared the cells." So 2 immediate thoughts occurred to me. The first one was, my foot is somewhere deep down my throat. How do I get it out? But the second one was, why is it that answers are always changing in science? How is it that something could have been an artifact and then not an artifact? Or I suppose a corollary question to that I get from teaching cell biology here is that why are completely new editions of textbooks being released every 3 years? Not only being released, but if you look into them, the various models they have in there for how things function are actually quite different. These actually are 4 of the 6 editions of what's one of the leading cell biology textbooks by Alberts et al. So why are they changing? Well, it could be that publishers just need more money. Maybe the textbook colors have to match the colors in vogue. But I realize perhaps a more serious possibility is that maybe scientists are correct. Maybe there really is such a thing as evolution. It's just evolution is happening so quickly that basic mechanisms in science are simply changing every 3 years, and thus they have to come out with a new textbook. Or A and B. So I became determined at this point to actually try to learn what is the secretory pathway? How does it function? If I ever stated it officially, I would say that I was doing it for the purpose of understanding fundamentally how does secretion work, because it's very important for understanding the body. If you ever asked me very quietly in the dark, I would tell you, it's to prove Mr. F wrong. So I started looking into the secretory pathway, and actually this is the work that I did for my graduate work. I worked on for part of my postdoc here, and actually still a major focus of work in my lab. So the secretory pathway was actually first studied for studying how a nerve cell sends signals to muscle. So I mentioned you secrete neurotransmitter. So what is the predominant theory for how neurotransmitter is released? Anyone? How is transmitter released from a nerve cell? Teachers can answer this also.
The impulse gets to the end of the cell, the neuron, and at that point the electrical potential triggers chemical release, which jumps the gap to the next one.
So the claim is that you have an electrical signal that comes down into the terminal, which stimulates the release then of the chemical signals. So that is the current model that they're trying to teach you in school. However, in fact, the dominant model that's only been around for like 50 years, the dominant model that's been around for over 1,800 years is actually quite different. It actually was first articulated by physicians in Alexandria, 300 BC. Basically, they said that when you've got in this case, it's being described here by René Descartes. Where it's still believed back in 1500s. Basically, they said that when fire gets a little too close to your foot, this results in pulling on this string, which opens a gate that allows animal spirits to flow. For almost 2,000 years, the dominant theory has been that you have flow of animal spirits down your nerve into the muscle, and it's this flow of spirits that results in the muscle growing, causing a contraction. This was completely accepted by every physician, every scientist who addressed it, until an experiment that was done where someone tried to measure the magnitude of the fluid flow. So what they did is they took a muscle and they put it inside of a jar filled with fluid that had a tiny thin tube on the top, such that when the animal spirits would flow into the muscle, you should see a change in the level of fluid on the top, and therefore you can measure the amount of animal spirits flowing in. The problem is, this never changed. You could have the muscle contract as much as you want, and there was no change. And in a single experiment, what had been the dominant theory for 1,800 years just went. Yours has only been around 50 years. Give it time. It'll go. So this was right around the time, this 1590, 1600, when Europeans were first exploring Africa, South America, and they were discovering all sorts of new things that at least Western Europeans weren't aware of, things like the electric eel. These things can generate a huge amount of electricity, and Europeans were fascinated by them, and they realized, oh, well, if electric eels can generate electricity, maybe our bodies are doing the same. And they proceeded to do all sorts of experiments to actually test these things. So scientists, you'll notice, are real slaves to various fads and fashions. So they would do experiments like this. They would, you can actually try this at home with your younger siblings. If you hang them up and you get a static electricity generator, you can actually show that the spark gets transmitted through the person. And so immediately this switched to saying there was an electrical signal that went not only through the body, but an electrical signal that went from the nerve to the muscle. This became a highly contentious field, particularly between older brothers and younger sisters, and led to one of the most famous fights ever in science. Debate would be too kind of a word, in which Luigi Galvani hypothesized that there was actually a real animal electricity in our bodies. Our bodies themselves would have electrical signals, as that gentleman in the back proposed. And the way he did it was by forming a loop of metal between part of a nerve and a muscle and showing he can get the muscle to contract. And he wrote a book called De Viribus, in which he said, here's the demonstration of the real endogenous intrinsic animal electricity. So Alessandro Volta, who read the book, found that he could easily reproduce the experiments, but after reproducing them, he proposed that it was because Galvani was using two different metals together here. It was at the junction of the two metals that he was artificially creating electricity. In other words, it's not that he was seeing an intrinsic electricity in the body, he was creating an external electricity that was causing the contraction in the muscle. So Galvani then responded by showing that he can then get contractions in the frog muscle using only a single metal. Volta, not to be outdone, then responded that it was necessary to dampen the contacts between the metal and the tissue to be able to get that contraction going on. So you can't just put the metal there, you need to wet it a little bit. And he said that it was at this junction between the metal and the solution that he was actually generating electricity. It wasn't actually the body itself that was generating electricity. And to demonstrate it, Volta invented a stack of metal and liquid, something called the voltaic pile, in which he showed he could generate electricity. Galvani then responded by showing if he only made a contact with a nerve going down to the muscle, that in of itself was enough to cause the contraction. So the question is, who won this debate? You could say that Galvani won, because he showed that you can stimulate the nerve even in the absence of using these mixtures of metals together. You could argue that Volta won, in that Galvani never demonstrated that the signal from the nerve to the muscle was electrical. He only showed that if he used an electrical signal, he can cause the contraction. Alternatively, you could argue that we all won, because in the process of this battle, Volta invented the voltaic pile, which we now call the battery. So the battery was a consequence of this debate between the two of them. So the issue remained, how are signals transferred from a nerve to a muscle? And one of the most visible experiments that's been done to that effect, the one that's quoted most frequently in the textbooks, was done by Otto Lowy, his identification of what's called vagus stuff. The experiment that Lowy did was the following. It was known that under certain conditions, if you stimulate a nerve, you could either have your heartbeat faster, or with another nerve, you can actually slow the heartbeat down. So what he did is that he stimulated this nerve in one frog so that the heart would slow down. He then collected the solution, bathing the heart, and added that solution to another heart, and then that heart rate slowed. Since there was no connection between the two, in other words, there was no electrical circuit, there was no wire, there couldn't be an electrical signal going to this. It would have to be a chemical signal. And for this work, Lowy received the Nobel Prize in 1936. There's only one minor caveat to this. The reason why you never do this experiment in classrooms, besides the fact that too many of us have watched E.T., is that, in fact, you can't reproduce this experiment. I've tried, it just doesn't work. It turns out, though, that now we have a good explanation for it, but for many years we didn't. It turns out that normally when you stimulate this nerve to release the chemical signal, your heart, as a way of regulating the amount of signal there, has various enzymes there that chew up the signal, so the signal doesn't stay around a long period of time. So it's released and then digested. Released, digested. Otto Lowy did his experiments during February and March when frogs were hibernating. And it turns out when they were hibernating, they had very little of the enzyme there to chew up this particular chemical molecule, which is why the experiments happened to work in his own hands. In fact, it got to the point where so many people could not reproduce his experiments that they would have him fly to places to demonstrate it, but they would insist that he had to stand 10 feet away from the experiment because they thought that maybe he would have some of that magic chemical stuff under his fingernails or contaminating things. So you'd actually have to stand at a distance as people reproduced the experiment to show that it actually could work. Perhaps the strongest experiment demonstrating that there was a chemical nature came from the work of Katz and Mulady. They took a very, very fine electrode for measuring electricity, and they moved it very close in to where a nerve was contacting a muscle. And they found when they brought that electrode very close in, they would observe little blips in the muscle. And in fact, if they stimulated the nerve, they would get a very large blip. And right around the same time, and what they did is they identified the molecule that they thought was being released. It turns out it was the same molecule, acetylcholine, that Otto Lowy had identified in the heart. They showed that if they puffed on small amounts of acetylcholine, they can get this same kind of reaction. And right around the same time they were doing this work, this was at the time that scientists at Rockefeller, starting in 1945, for the first time used the electron microscope to look at biological material. In fact, it was for that work that Pallotti, together with Albert Claude and Christian de Duve, all here at the Rockefeller, got the Nobel Prize. And when they looked at nerve preparations, I actually think this may be a sample from Keith Porter, what they observed is that inside of the endings of all of the nerve cells, they would see these little round packets. Here's another example from a frog junction, these little round packets. And it turns out if you stimulate the nerve, well, actually you have to stimulate it really, really, really hard, you can end up catching images that look like this. This is from the work of John Heuser and Tom Rees. Things that look like a vesicle that's in the process of releasing its contents. So it's for this work that in 1970, Bernard Katz got the Nobel Prize. Their demonstration of these little blips was in 1954. And right after they presented this, there was immediate consensus in the scientific community that all transmission was chemical. It was a result of these little packets releasing chemical transmitters. Just 5 years later, First Pan and Potter demonstrated for the first time that in the crayfish, all signals that go from the nerve to the muscle are actually being transmitted electrically. So after 200 years of fighting over this issue, the scientific community finally reached a consensus, which is, this is the consensus you're taught nowadays, yet just 5 years later was the first demonstration that in the crayfish, signals between the nerve and the muscle are electrical. And in fact, we now know today that many of the connections in our own brains use electrical signaling. So for example, in your brainstem, there's a section called the inferior olive. The inferior olive produces nerve cells that go up to your cerebellum, which is in the back, that coordinate motor coordination. All of the cells sitting in this thing called the inferior olive are coupled to each other electrically. And they're all firing together in unison. It's those electrical connections that keep them firing in unison. Your teachers could actually test this out. It turns out that one very effective way of inhibiting these electrical junctions between the nerve cells is with ethanol. And so as you add more ethanol, you inhibit the connections, which affects the firing to the cerebellum, and you disrupt motor coordination. When you get to college, you can do a lab on it. So the conclusions that were reached is that transmitters stored in membrane, that's a typo, membrane-bound spheres we call vesicles. Vesicles fuse to the surface to release their cargo. Yet the most recent article I know about on synaptic transmission in Scientific American actually completely rejected the vesicle hypothesis. And the question is why? Well, it turns out there were a number of problems with that result I just showed you from the electron microscopy, the one that showed this little shape here. So one problem is that with electron microscopy, you're just catching a cell at a single point in time. There's no sense of the time course. So for example, you could be seeing a vesicle here that was in the process of releasing stuff, or maybe what you're catching is something much later on, taking things back up into the cell. The little image I showed you, is that something going out or coming back in? Without being able to follow it live in a cell, you can't tell which way it's going. Furthermore, the things that look like the Greek letter omega, shaped like this, that were seen, they were only seen when you really stimulated the cell really, really hard. Maybe it was just some weird pathology of the cell. So what was needed was a way to see the vesicles, watch them come up to the surface, the ability to see, do they fuse? So the question is how to see things in a cell. So one of the best ways of following things in a cell is to make the cargo fluorescent, or make the molecules you want to see fluorescent. There are two problems with this. One is how do you make cargo? So in this case, it's protein we're going to look at going out in these vesicles. How do you make it fluorescent? And how do you see something that is small? When I mean small, 30 to 300 nanometers. That's smaller than the wavelength of light. So the first question, making something fluorescent, has become easy in recent years. One thing you can do is you can purify what you want to study, and you can chemically link to it something that is fluorescent, and therefore make your molecule fluorescent. The other way you can do it is to take the DNA that encodes your protein and attach to it the DNA that encodes a fluorescent protein from jellyfish. So for example, the green fluorescent protein found in this jellyfish. And in fact, it was for this kind of technology that 2 weeks ago, Shimomura, Chalfie, and Chen shared the Nobel Prize for developing this technology of making proteins fluorescent by fusing them to the genes encoding the green fluorescent protein. But how do you see things that are small? So this is a non-trivial problem. We use light as our detector, and therefore light behaves like a wave, and the size of a wavelength of light affects our sensitivity. So let's say if I was blind and I was walking with a cane, I could use the cane to find rough features. Where is there a barrier? Where is there a curb? But if the cane is large, it can't pick up the fine details of Braille. If you wanted to pick up the fine details of Braille, you'd have to use something not the thickness of a cane, but something much thinner. Either a toothpick or even the fine sensory endings in your finger. So the bigger the probe is that you're using, the less sensitive it is to very small things. If we're using light that's on the order of 500 to 600 nanometers, we can't really resolve things that are much, much smaller than the wavelength of light. Yet these vesicles that I wanted to see, the ones I wanted to see coming up to the surface, fusing and releasing, were much smaller than that. So I struggled with this problem for a while, and I finally did what I suppose most of you would do in a case like this. I packed up my family and I went on a vacation to France. More specifically, I went to the Collage de France. Actually, not packing up my family to go on vacation. I went on a vacation to see family because it was specifically here at the Collage de France that my wife's brother was working, where he was studying polymer movement. So he'd gotten his Ph.D. in optics, and he was taking advantage of his Ph.D. in optics to try to study polymers moving in solution. And he was working in the lab of P.G. Dejean, who is the world's absolute expert in polymer movement. And the specific question that Kalman wanted to go after was, do polymers move the same out in bulk solution? Polymers like these long chains of things. You can imagine a DNA molecule is a polymer. A long thing of sugars is a polymer. Do they move the same in bulk solution as they do near a surface? And when I'd spoken to him in advance, I said, well, how are you asking this question? He said, well, I'm just watching them. By a microscope, I'm making them fluorescent. I said, well, when you said near a surface, like how near to the surface? And he said he was looking on the order of tens of nanometers. And that didn't make sense to me because that's less than a tenth of the wavelength of light. So I wanted to go visit him to see how they were actually doing it. And it turns out that what he was doing was taking advantage of a little trick, at least the way he described it to me, is of bringing in a laser beam through glass at an angle so steep that it just reflected back. And he said that if you come in at a steep enough angle so that all the light reflects back in, you have no light being propagated this way, but it turns out there's a very, very thin area of excitation right above the interface between the glass and the water that decays over a distance of roughly a tenth of the wavelength of light. So this sounded sort of like magic to me. How can you create this little thing of excitation that is fractions of the wavelength of light right above this glass? And it doesn't cost you any energy. There's no energy propagated that way. But Kalman was working in the lab of Dejean, who actually the following year got the Nobel Prize in physics by himself, solely for discovering these kinds of methods for studying polymer movements. I figured, maybe there's something to it. So I decided I would try it out. So this is how the technique actually works. What you do is you take a laser beam going through glass up into water, and if you change the angle of your laser beam as it comes up into the water, it refracts. But if you get above a certain critical angle, it completely reflects internally, leaving just a thin field of excitation over the surface. And this thin field of excitation, you can leave aside the math for the moment. The critical thing is that it decays over a distance of about a tenth of the wavelength of light. So I decided to try to set this up in my lab and ask, can we use this to actually see small vesicles coming to the surface and releasing their contents? And we took advantage of some microscopes where you can bring a laser beam in from on the outside at such a steep angle, it reflects, and it should create this very, very thin field there. This is the actual first microscope we set up in the lab. Most of it not only was, but is still held together by tape, bubble gum, styrofoam, but it works. And what we decided to do was label some cargo inside of the secretory pathway with a green fluorescent protein. And our hypothesis was that if vesicles were coming to the surface of the cell and releasing their contents, what we should see is that as each vesicle reaches the surface, it should undergo an explosion as it released its contents. So this is what the cell looked like if we just used sort of standard microscopy. This is what it looked like if we used this technique of what is referred to as total internal reflection, meaning that the light's being totally reflected internally. In this case, we were only seeing those vesicles that were within tens of nanometers of the surface. So if you then look at the cell, and this is actually the image looking at human growth hormone done by Jyoti Jaiswal in my lab. And could we dim these lights up here? Just the ones above the screen, thank you. In fact, what we observed was as these vesicles came up to the surface, we see them undergo these beautiful explosions. And I remember the moment I first did this experiment, I was absolutely excited. It was exactly what I had predicted, exactly what I expected to see. In fact, I was so excited, I got a second dish of cells. Now, you're always afraid it's a fluke of the... It worked on the second dish of cells, and then the third dish of cells. And then I decided, let's try different cell types. I went to a second, third, fourth cell type. And it got to the point where I think I had done about 12 experiments in a row, and I started to get nervous. I don't know what your experience is like in classroom laboratories, but my experience at the bench is that there are no experiments that work each and every time. In fact, let me state it more strongly. Most experiments fail, and they fail for a variety of reasons. Yet here, every single time I did this experiment, it worked. In fact, even today, if you do this experiment the same way, you will see these vesicles coming up to the surface. And so there are a few things that started to bother me about it. So here's a close-up of one of those things that underwent a burst over here. One being is if I then focused in on one area of the cell, and then I moved over to where I hadn't been imaging, it was only where I was examining the cell that the vesicles had released their contents. So all the other vesicles here were waiting for me to look at them before they would release their contents, which was sort of cooperative of them. And in fact, I can then go from dish to dish, and only in those areas where I zoomed in would the vesicles release their contents. So now there were two problems. There was an experiment that worked every single time, and they waited for me to actually... And after actually a very long time, too long for me to actually tell you because it's embarrassing, I suddenly realized that what I had hypothesized in advance is that I should see a vesicle come up to the surface and release its contents, giving that explosion. Maybe what I was seeing was a vesicle coming up near to the surface where it was being hit by my laser beam and it was exploding. It wasn't funny back then. This is what I was referring to before, that you can have a hypothesis and get a result that's consistent with the hypothesis, but in fact there can be other hypotheses, other models that make the same predictions. In what took me about another 2 years to figure out how to distinguish between them, I came up with the following idea. Instead of labeling cargo on the inside of the secretory pathway, to label a membrane protein with a green fluorescent protein. So what would that do for us? Well, if this was coming forward and fusing, it should get brighter as you get closer, and then dimmer as everything diffused away into the membrane. If this was coming forward and then lysing, it should get brighter as it gets closer, and then dimmer as everything diffuses away. So that wouldn't help. But if these things were truly fusing to the surface, all of these membrane proteins should be getting delivered to the surface, which means if we now summed up all of the fluorescence over the surface, as each vesicle came there, the total fluorescence should go up, but it should stay up because all of the membrane proteins have been delivered to the plasma membrane. We've delivered new surface to the area of the cell, and we can count up those fluorophores. Whereas if this moved forward and lysed, you should lose that fluorescence back into the cell. The other way we can distinguish it, if we just measure the area covered by this fluorescence, it should spread out. And we understand how it should spread out. We understand the rules of diffusion. The area should increase linearly with time. In fact, that gives us information about the diffusion constant, the speed at which this protein moves. Whereas if this was lysing, it should diffuse out into the 3 dimensions of the cell much more quickly. So here's a close-up now of a vesicle where we've labeled one of the membrane proteins. And you'll see it moving along now. This is an elongated vesicle. In the very bottom surface of the cell, you'll see it pause here and then fuse. The critical thing now is to distinguish, is this due to the thing breaking open, being zapped by my lightsaber, or is it actually fusing into the membrane? So if we actually now quantified our fluorescence, here in black was the total fluorescence. It goes up and stays up. it's delivered all of those membrane proteins to the plasma membrane. Here's an example from a smaller vesicle. It goes up and stays up. If we now measure the rate at which it spreads out laterally, it spreads out exactly, the area increases linearly with time, exactly what you expect from something diffusing out into the membrane. So now we had a way of distinguishing between lysis and fusion. And then when we started looking, we found some of these, when they reached the surface, they delivered all of their contents in a single step, like that one. Yet other ones, when they reached the surface, would release their contents in more than one step. So if you watch, you'll see this one start to fuse right over here, releasing about half the contents, and then the rest of it fuses over there. In fact, we saw a large number of these vesicles that would fuse a number of times to release their contents. So this was now a slide from about 15, 20 minutes ago where I told you what were some of the kinds of things we worry about. And so the key things were, are there other hypotheses that make predictions that are equally consistent with our hypothesis? That's our constant fear with everything we do in science. And then, is the result a consequence of how we did the experiment? In other words, were we causing lysis of the vesicles or causing something else other than what we intended to do? So my initial results, those fusion, they were completely consistent with my hypothesis of fusion. However, it does not mean that my hypothesis was proved. In fact, we know that initial hypothesis was wrong because those first events of release from the cell were an artifact. There was another hypothesis, in this case, that the vesicles were being broken open by the light excitation. And so in those very first experiments I showed you, the predictions of the hypothesis for release were exactly the same as the predictions for the hypothesis saying there was lysis. So the take-home lessons. One, you should try to test every hypothesis with different techniques. So every technique is going to have its own set of potential artifacts, its own set of potential assumptions. However, it's less likely that if you use different techniques, that they'll share the same set of assumptions or potential artifacts. This is also the advantage of having a number of different labs trying to address the same question. Very often, I've heard Congress people argue, well, why should we fund two different labs to be going after the same kind of question? Why not have one lab do this question, another lab do the other question? It turns out that every lab takes a different approach to the same problem, and each one of these approaches has their own set of assumptions and limitations. And sort of the third take-home lesson is that from observing things, you'll see things you might never had hypothesized. In this case, the vesicles, they're releasing their contents in more than one step. So very often in science, and particularly science education, we emphasize the role of having a hypothesis in driving science. However, very often it's the observation that's absolutely essential for the hypotheses. So for example, Darwin never could have had his hypothesis about evolution if he hadn't first spent time traveling around observing many different animals. And these kinds of observations end up being the foundations for all the hypotheses. And hypotheses themselves will come and go with time, but it's the observations, if done right, that remain static. So why is imaging having such an impact today? There are two ways to look at it. First, advances in cameras have increased sensitivity incredibly. They've gotten to the point now where we can watch single molecules, and I'll give you an example of that a little later on. There are advances in digital electronics that have gotten to the point now where we can process thousands of images a second, which means we can follow things very quickly inside of cells. There are advances in chemistry that allow us to bind fluorophores to various proteins, molecules, and follow them in the cell. There are also advances in chemistry. I'll give you an example where we can actually activate the fluorescence very specifically wherever and whenever we want to. There are advances in molecular biology that allow us to modify proteins in such a way to make them fluorescent. But all of these things just say, why has imaging become easier to do? They don't really answer the question of why imaging is having such an impact on science. And for that, actually, we have a different set of answers. I think it comes down to four things. First, imaging allows us to study living systems. Imaging allows us to stimulate or activate different pathways. This is an extremely important point that I'll spend quite a bit of time on. Imaging gives us information about the diversity of a system. And finally, imaging allows us to examine a multitude of sizes simultaneously, from single molecules to whole cells. So first, living systems. There's certain things you can learn from living systems. Let's say you want to understand how the circulation works. There's certain things that you can gather from going down to the morgue and looking at cadavers there, which are very different from what you can get from actually taking a living human being and actually following the heartbeat, following the blood flow through the body. So there are things you can learn just from observing living things that you can't get from a fixed sample. So let me give you a more specific example of blood flow. And that is, let's ask the question, does the pattern of blood flow in a heart actually affect development of a heart? So it's been known that if you take isolated heart cells, you put them down in a dish, and you just change the rate at which you flow solution over them, you'll change the expression of different genes in those heart cells. The question is, does this happen in a living heart? So the way this was done was to image blood flow in the heart inside of a living zebrafish embryo. And this was done not only by labeling the blood cells with fluorescence, but you can actually label the heart cells in such a way that you can arrange so that you have, as you get changes in gene expression, you get changes in the expression of different fluorescent proteins. You can actually have the cells change not only intensity of fluorescence, but color of fluorescence as you change the expression of different genes. So this is an actual image inside of a living zebrafish heart. This is from the work of Scott Fraser and colleagues. And one of the things they noticed is that first is a very, very high rate of flow in the heart, much higher than they thought, giving real high shear forces, which is much greater than they had previously expected. If they then altered the blood flow, which you can do by sort of pinching down on the blood vessels a little bit, you can actually change gene expression. So by increasing or decreasing the rate of blood flow, you actually alter gene expression in the different heart cells. And in fact, they would alter, so the first level of muscle cells were different from the second level, different from the third level. What you needed to do is actually look in the living heart and see all the different levels of cells and the gene expression in each of the separate cells to resolve these differences. So this could not have been determined by genetic analysis. So in other words, if you now looked at total gene expression, you'd be averaging total gene expression for the entire heart. What they were seeing were very fine differences from one cell to the next as a function of this blood flow. This could not have been determined by biochemistry. So biochemists, if they wanted to see a change in expression of certain proteins or other molecule, they would just take the heart, grind it up, and say, okay, how much of this protein do we have, how much of that protein? It was only by imaging where you can see everything not only on a cell-by-cell basis, but even on a finer level in the cells, can you resolve these differences in expression. So another area where you can use living things to learn something about the biology and blood flow comes from the question of what happens when a blood vessel is blocked. So blockage of blood vessels is unfortunately not an infrequent event, and it causes severe problems in a number of areas. For example, in the heart, you lose blood flow to an area of the heart, you have a heart attack. To the brain, you have a stroke when you lose blood flow. In the eye, so one of the severe problems, for example, in diabetes is loss of blood flow to certain areas of the eye or the periphery. It's a major cause of vision loss in not only diabetes, but age-related macular degeneration. Any kind of transient block in the vasculature causes loss in vision. So the way we can study this now is by actually imaging blood flow in the back of the retina in a living animal. It's actually imaging right through the eye. So here's an image taken on the back of the retina. This is actually from the lab of Martin Friedlander, who also used to be a postdoc with Gunter Blobel. And what they found is that imaging the blood flow in the back of the retina was absolutely essential for understanding normal development of the eye, progression of disease, the progress of different experimental treatments they were using. So Marty himself is an ophthalmologist. He's a clinician who's studying various ways of treating problems of visual degeneration. So he's been using the system to study development of blood vessels in the retina, and he's been using it as a test system for compounds now that will either facilitate blood flow, so when you lose blood flow, ways of bringing it back in, or ways of blocking blood vessel growth. That's one of the problems in diabetes, when you get extra blood vessels growing in there that will block the vision. And most recently, they've been using this now to use stem cells from the bone of the animals now to repopulate the retina to allow new blood vessels to grow in. This is something that could not have been determined by genetics or biochemistry. They were only able to examine this question by using imaging, in this case in a living animal. Let me give you an example of how imaging helps with living cells from the work of secretion. So we mentioned that secretion involves release of material to the outside, internalizing material from the surface coming back in. So we know many of the molecules that are involved in this process, and they go by all sorts of fancy names that you can read up about. They're called snares and synaptotagmin. That's not really relevant to the moment. The critical thing we want to know is, what are these molecules doing? What is happening when things are working properly? And for that matter, what happens when they're not working properly, and what can be done to correct them? So there are two ways you could approach secretion. One of which is to actually look inside of a living cell and label the different components and ask who is coming in when, who is leaving when. The other way is to purify each of the components involved and actually try to put them back together one by one and build up the machinery for doing this outside of the cell. So I already showed you how we can follow secretion in the living cell, and we've used this for a number of purposes. For example, we've now labeled individual components with different fluorescent proteins, and we've followed them moving, asking when do they come, when do they leave. It turned out certain key proteins that we thought were involved in doing the fusion actually left before the fusion step occurred. I'll give you some examples of that. But we can also use this to approach some important clinical problems. For example, in diabetes, we can watch single packets now releasing insulin, or we can watch single membrane-bound packets carrying the sugar transporter, GLUT4, that gets delivered to the surface only when insulin's raised. And I have some videos of that I can show you after the talk for anybody who's interested. What this allows us to do now is study the effects of, for example, single mutations that are found in diabetes on this process, or test for various therapeutic treatments of it. But we can also apply it to other clinical areas. So it turns out this process of a vesicle coming up to the surface and fusing is absolutely essential for fixing rips and tears in the surface of a cell. So whenever you move, your muscles are constantly getting ripped and torn, and this process of membrane coming up and fusing is what's repairing those tears in the membrane. So it turns out that many forms of muscular dystrophy result in point mutations in proteins that are essential for this process to occur. So the muscle can now no longer repair the tears in its surface. We can actually image how this process occurs, and actually Jyoti Jaiswal in my lab, who started off with us studying secretion, is now devoting all of his effort to the specific question of how does membrane repair occur and what happens in various forms of muscular dystrophy. So biochemistry and genetics were absolutely essential for us to identify the molecules involved. However, it's through imaging that we can now start asking the question of how are these different molecules functioning. So I mentioned one way to address this is look in the living cell. The other way is to test our knowledge by asking, do we understand this system well enough that we can take each of these separate components and put them back together and build up the machinery outside of the cell? And if we really understand this right, we should be able to put them together and build our own secretion apparatus. So what I'm going to tell you right now is some work using imaging from the work of Marina Fix in the lab, which she did together with Jyoti, who I mentioned already, and Josh, who you'll meet in a few moments. And what she tried to do was the following. She asked, can I form just a flat membrane, just like the one you have on the surface of a cell, form a round membrane-bound packet of lipid, and then test various proteins or various other components and ask, what are the key things I need to build up this machinery and get it to work? So what she did, she made these little membrane-bound vesicles. In this case, she added a fluorophore to them, and she made them containing only one protein in the vesicle, and there were only two proteins in what became the equivalent of the plasma membrane. And right here at the X is where she stimulated fusion. And what you should start seeing are bright flashes there. Can you tell? I can't tell with these lights. Can you see the flashes on the screen? So actually, if you look here at the quantification, if you look, for example, at this one over here, this one got brighter, and then its fluorescence spread out. Once it fused, all of its fluorophores were diffusing into the plane of the membrane. She was actually able to reconstruct the process of a vesicle fusing to the membrane. Now, a critical question is, how do we judge whether this is really reproducing what happens in the cell? So there are many kinds of reaction that can occur. So for example, let's say we want to understand membrane fusion. I could take a garbage bag, fill it up with soap bubbles, and then if I crunch the garbage bag down, some of these soap bubbles will fuse to each other. That doesn't mean that that fusion I'm getting of soap bubbles crunching together is the same as the kind of fusion that's occurring where a vesicle is fusing to the surface of a cell. What we want to know is this kind of process of a vesicle fusing down, is that the same as the one in a cell? So one important thing is that to see whether it's the biological mechanism is to realize that what biology really cares about is rates. And we want to know, is the vesicle fusing in a manner that tells us something about what happens in the cell? So the kind of tests we can do is ask, does it use the right molecules? So it turns out if you do not use these very specific molecules used in fusion, it didn't happen. Does it occur at the kinds of rates that occur in a cell? Well, these were happening in milliseconds, sort of about the rate of what you would get in a cell, but not quite fast enough, but getting close. Does it occur with the kind of efficiency? In other words, if you're seeing one vesicle out of 1,000,000 fusing, that's not very good if inside of the cell you're getting 1 out of 100. Does it show the same kinds of regulation as observed in a cell? Or does it show the same kind of sensitivity to various toxins? So for example, botulinum toxin or tetanus toxin causes an arrest of motion because tetanus toxin actually cleaves one of those 2 key proteins that she was using that's required for fusion. If you get tetanus toxin in you and it digests this one protein, it's called a snare, what happens is the vesicle can no longer fuse, you can no longer send a signal from your nerve to your muscle to move, and thus you get tetanus. So it turns out the reactions that she was studying had the same sensitivity to toxins, tetanus toxin, botulinum toxin. We have all sorts of fun toys in the lab if you're interested. It had the same kinds of regulation and the same kinds of efficiency. So I mentioned that one of the reasons why I think imaging is having such impact is that you can use it in living systems, which means you can learn things that require a system to still be alive. You can learn things that are not available from static images, but also imaging is facilitating the development of various diagnostic techniques that are non-invasive. So until some of these imaging techniques were developed, let's say 15, 20 years ago, if the doctors suspected that you had an abnormality in your intestine or they thought you had a tumor, they had no option but to cut you open and see, is it there or not? Nowadays, you can just go in for a scan and see, is the intestine okay? Is there a tumor there or not? And I think most of you, if you had a choice between a surgeon going in and sticking their hands inside of your abdomen versus going in for a 20-minute scan, I think I know which one you would choose. And imaging is allowing the development of these kinds of techniques. So just before we break, let me briefly mention the second reason why I think imaging is having such an impact, and that it allows us to stimulate or activate different pathways. And I wanted to just give you a brief example from the work of my colleague here at Rockefeller, Tom Muir. Tom is a chemist, and what he's been asking is, can we use light to regulate the fluorescence of a protein and regulate its activity? And the model system he's been looking at, you could ignore a lot of the proteins around here, is one in which when you add in something outside of a cell to stimulate it, there's a signaling molecule inside which is normally on its own, but it then forms an oligomer. 3 of them get together into a trimer. And it's that trimer, which is the important signaling molecule, which can now go into the nucleus and stimulate a response. And what he wants to know is, can we actually trigger both the activity of this protein with light and trigger its being fluorescent so we can then follow it? And the trick he used was to purify one of these, add on a little molecule that made it fluorescent, but then added on a second molecule in such a way that that second molecule would absorb all the fluorescence, it would quench all the fluorescence. So as long as this molecule here was attached, you could no longer see the green fluorescence. Furthermore, this molecule is attached in such a way that it blocked this thing from forming a trimer, getting together. And since he's a chemist, he was able to design it in such a way that when he shined light of a particular wavelength, what it would do, it would cause this group to fall off. Once this group fell off, this was now fluorescent. This can now assemble, go in the nucleus, and start triggering. So here's a picture of a cell in which the cell has been loaded with these molecules prior to being activated. And then once I click on this, what you'll see is they've now flashed light on it to remove the group that's quenching it. The molecules now turn green, assemble, and they can go into the nucleus. This is just a demonstration on the level of the whole cell, but in fact, what you can do is you can just stimulate one very small area at a time and locally activate proteins in one part of the cell or another. So after the break, I'm going to go into the question of the diversity, really the advantages of studying single molecules. So we were addressing the question of why is imaging having such an impact today? And I think that actually one of the major reasons why imaging is having such an impact is that it allows us to follow each of the members of a population and get a sense of the various variations or the diversity in a system, whether it's looking at single molecules, single machines in a cell, or single cells. And I'm going to give you a number of examples of how that's really given us tremendous insights into biology that weren't available before. So why study individual events? There are going to be 4 reasons that I'm going to give you. First of all, the macroscopic behavior just reveals the average. So let's say if you had a population that existed in 2 separate states, if you took the average, you may come up with some value in between that actually none of the members of the population actually have. I'll give you an example in a moment. The other reason for looking at individuals is that it may be that the average is telling you about the dominant population, but very often in biology what we care about are very transient events, things that happen just for a moment or that are very localized in space or time. If you just look at the average, you may miss those. So for example, there may be an enzyme turned on very, very briefly. So for example, if you've ever seen pictures of cells going through the cell cycle, there are some images of them before. It turns out there are certain proteins that get a particular modification on them for just a few minutes during the cell cycle. If you just took the average population in a dish, you'd say, nah, that protein's never modified because it lasts for only 5 minutes out of, let's say, a 48-hour period. You'd never pick it up. So a lot of the things we care about in biology are very transient events, and thus you can get that only from looking at individuals. A third reason, and I'm going to give you more examples of each of these, is that from looking at single individual events, we can get information about the time course with which things are happening that you can't get from just looking at the average population. In the case of the cell cycle, if you followed an individual over time, you can see there's a certain point where it's right when the nucleus is about to break down where you suddenly discover certain modifications are occurring. And you can get a sense of the order of events that you can't get from just measuring an average population. And finally, I'll give you some examples of a number of cases where if you just look at the population and you take measurements, you come up with a number of explanations for what you're seeing, but you can't resolve between the models. But if you look at single individuals, you can resolve between the models. So let's go through these in more detail. The first one. The first example is, I said, what happens if you have a population that's actually in 2 separate groups? So let's say I'm a biologist. I want to study human chromosomes. And as many of you may know, all humans have X chromosomes, but a certain subset of humans have Y chromosomes. And so let's say I want to figure out, okay, well, which human, how many Y chromosomes does a human have? So one way I could do it is I can take, let's say, maybe, 200 volunteers from the frontier. I can chop off your arms, mash them up, digest them, solubilize them, run the chromosomes out on a gel. And let's say from all of you, I think I'd come up with the estimate that the average human has half of a Y chromosome. Now, there's an obvious flaw in there. If you're male, you have 1 Y chromosome. If you're female, you have none. Even in New York City, there are no humans that have half of a Y chromosome. And I've seen a lot of strange things in New York. So here's a case where by taking the average, you get a misleading result. There are no humans with half of a Y. You either have 1 or you have none. So there's a case. Let me give you another example. Let's say we want to understand how proteins move. The way we usually approach it is what we'll do is we'll take our particular protein we want to study, we'll make it fluorescent, and the way we'll study the protein movement, since it's sort of hard to see the individuals, what most people will do is shine a very bright laser beam on a small area, destroy all the fluorescence in that area, and look at the rate at which fluorescence comes back into that area as an indication of how a protein's moving. Well, the problem with that, let's say you wanted to understand, let's say you want to know how quickly does a human walk. It'd be the equivalent of, let's say, going to Times Square in the middle of the day and using some kind of freeze ray to destroy everyone within a 2-block area and just measuring the rate at which people came back in. The problem with that is that you're not getting the rate at which an average person walks, because some of those people will be stopping to look in the windows, some of them are going to be stopping to borrow other people's wallets, some of them are going to be they're not just walking at a steady rate. And it was a similar problem for studying protein movement. So the way we tried to get around it instead was to put tags on each individual protein, because if you followed a single individual, just measured the rate at which they moved around. And we did this with a protein with which people had done the standard approach of measuring the average rate of movement. And the problem we had is that, first of all, our measurement didn't match. We got a very different value. But it's not so much that we got a different value from them, we got a different value from ourselves. In other words, when we followed the protein, one moment it was moving very fast, and then it wasn't moving. And then it was moving fast. And we realized we were getting many different values for what they call the diffusion constant, the rate at which a protein moves. And this was very disturbing. I thought there was something wrong first in my technique. Maybe there was something wrong in how I analyzed the data. Maybe there was something wrong in our imaging. Maybe what I was really seeing was just bumps due to my walking around, which was jostling the microscope, which was causing the particles to move faster and slower. But in fact, what we found over time, when we repeated this over and over, is that this sort of made sense. Because if we just did the same experiment by putting our proteins into like a fake cell, like just a solution, they behaved normally. When we looked at them back in the cell, again, we got very different values for the rate of movement. But that makes sense if you think about it. The proteins were going along, and they would hit barriers, or they would stop and they would bind onto something, or they would stop and act on something. There's no reason to think that the protein was always moving at a constant rate. And the flaw of the standard approach of bleaching out an area and measuring the rate at which things move in is that it assumes that all proteins are moving at the same rate and the same time. It's an average value. And that average value for movement, in fact, didn't exist for any protein. They either were moving fast or not moving. Fast or not moving. The average we got, which was in between, wasn't an accurate reflection. So actually, the sort of important point, which if anyone can find a better way of wording this, I'd appreciate it. Basically, the mean is meaningless for knowing the rate at which a molecule moves. In fact, for a lot of phenomena, I would say the mean is meaningless, but there must be a better wording for that. So the second thing, I said the average population may in fact represent the dominant population, but what we often care about in biology are transient events, things that are very localized in space or time. And so I'm going to give you an example from some of our work, not so much on the process of a cell sending things back out, but taking things back in, the process of internalization. And so that process of internalization, which is also called endocytosis, is used for bringing nutrients, for bringing signaling molecules, and then recycling various components on the surface. And with time, cells will get damaged, proteins that are being hit by enzymes, things in the environment. And think of the cells in your stomach, right? They've now got in them 3 or 4 slices of Domino's pizza, soda. If you think of the trauma they're being exposed to, a lot of these proteins on the surface need to get recycled. They need to be brought back in and replaced. Sometimes you want to shut off the activity of proteins on the surface. So, for example, cells that are involved in digestion, or cells, for example, in your kidney that are involved in secretion, when you have a load of acid due to some things you may have eaten, let's say the pepperoni on the pizza, the way they respond is by actually putting into the membrane or taking out various proteins that will pump acid across the membrane. And they very actively regulate this process. So this is also the pathway that's used for various pathogens to enter the cell, which is another reason why we study it. So most viruses and bacteria use this as a way of getting access into the cell. So the question is, how would you go about studying this process of internalization? Now, many proteins have been identified that have been involved in this process. And the way they've been identified is that people have mutated cells and mutated organisms that are now defective in taking things up. So there's a large number of proteins involved. The problem is knowing what are each of these proteins doing? How are they working together? So the question is, how do they take up molecules? So one approach was now learning from the work we did on secretion. And actually, let me just divert for one moment and say that plagiarism in science is unfortunately maligned and should be honored. Especially since they're videotaping it there. I think I better word this very carefully. It's very rare in science. In fact, I'm very hard-pressed to think of an example where someone comes up with an idea totally de novo. In almost every case, what happens is we build upon something that has been done before. Or what will happen is someone will identify a technique that's very useful in one discipline that they can then use in another discipline. For example, my brother-in-law was using this technique that apparently was well known in physics for looking at things right near a surface. Well, it was very useful for studying things in biology. Gunter Blobel became famous because in part what he did was use the techniques that had been developed for studying metabolism, of breaking a cell apart, isolating out all the enzymes, but then applying it to a process that affected transport of membranes in a cell, showed that you can actually dissect apart the steps of membrane transport in a cell and transport proteins by breaking a cell apart and putting it back together. So he borrowed from one discipline and applied it into a new discipline to identify a whole new set of questions. So similar for endocytosis, we can build upon the kinds of approaches, endocytosis taking things in, build upon the same kind of approaches used for putting things out of a cell. So we can study this process inside the living cell, and we can do this, for example, by genetically modifying the various molecules so they fluoresce, and follow their movement. Ask, when do they come in? When do they leave? We can take advantage of genetics and to eliminate particular molecules and see how it affects the process. The second approach we can take is to try to build up this process outside of the cell. For today, I'm going to give you examples of the first approach, studying it inside of the living cell. So the work I'm going to tell you about is looking at one very particular pathway for internalizing things. It's a process called clathrin-mediated endocytosis. So it's a process of internalization. It turns out the cell uses quite a few pathways for bringing things in. This is one of the most predominant ones, and it's mainly identified by the use of this molecule clathrin on the surface, thus its name. And the approach that was taken, this is the work of Josh Rappaport, was in the lab. And actually, this picture is rather typical of Josh. For those of you in the back who can't see, this says area closed to public. Josh sort of goes where his intuition takes him, independent of what reviewers or signs may say. He also tends to like to take a difficult approach, but don't let that really fool you. The picture was actually taken on the side and was turned. So the approach that he took was to try to fuse the DNA that encodes various molecules that are involved for internalization to different colored fusion proteins to follow them moving around. So I mentioned one of the key molecules in this process of bringing things in was this molecule called clathrin. And what he observed was that these molecules were found in spots on the surface of the cell. And if you looked at these spots, some of them, such as this one, you can see that they're moving them across the surface of the cell. Other molecules would disappear. So if you look at this one spot over here, you can see it disappearing. So about 15% of these spots disappeared in a minute. So 15% in a minute means roughly 0.25% of them were disappearing per second. And as he was studying this under the microscope, he had a camera going, taking about 25 pictures per second, which meant that he was getting something on the order of 0.01% of them disappearing in any particular frame. So we're talking about 1 out of 10,000 of them disappearing per frame. So this was an extremely rare event. Oh, and actually, I should digress for a moment here, sort of with respect to Bonnie Kaiser. Bushra Taha, who's here, who's actually an author on these papers, was actually a high school student at the Dalton School who had signed up in Bonnie's summer internship program, the one for which she has these little notices outside. And Buscher had come into the lab and worked and actually published 2 papers on this work. So seeing just, 1 out of 10,000 molecules going in was a little disturbing. It's sort of an extremely rare event. But what Josh noticed is that in addition to seeing clathrin disappear, if you looked at another molecule involved in internalization, epsin, whenever you saw clathrin disappear, epsin disappeared at exactly the same time and the same place. So if you look over here, when this particular spot of clathrin disappeared, epsin that was in the same position disappeared in the same time and the same rate. Or if you looked at a cargo molecule, it turns out transferrin is involved in bringing iron into your cells. It's absolutely essential for you to stay alive. Transferrin, when this one particular spot of transferrin molecules disappears into the cell, the clathrin that's in the same position disappears in. So seeing the clathrin disappear from the surface is a rare event, a 1 in 10,000 event. For epsin, it's rare. For transferrin, it's rare. But for all of them, when one goes, the other goes. And so it gets to the point where if you're going to have 4, 5, 6 different markers for this vesicle all moving into the cell at the same time, it's almost impossible for all of them to be happening together unless they're really being internalized. It's not just sort of a statistical fluke. And it's something you have to be careful of when you study rare events, which is something we'll return to. So we decided this would be our marker for internalization. If we saw all of the components of this vesicle get together at once and disappear together at once, we were saying, okay, we're looking at the cell internalizing things. So then we could start looking at the dynamics of the molecules. The only problem was our experiments were fooling us. And this is sort of a classic example of how these kinds of experiments can be misleading. So it turns out one of the key molecules that we were studying for internalization is a complex called AP2. It's known as an adapter protein. This is what's supposed to be one of the key proteins involved in internalizing things with clathrin. And in fact, when we looked at the localization of clathrin and AP2, it was actually pretty good. So every place where you saw clathrin, pretty much you saw AP2. Pretty much everywhere you saw AP2, you see clathrin. You can see the overlap here in yellow. And for anyone who is colorblind, I apologize for the choice of colors, but Josh Rappaport is colorblind and he insists on using red and green even though he can't see the difference. If any of you are colorblind, perhaps you can explain to me why he would do that. Or Josh, if you're listening at home. In fact, you can quantify this. You can go into the computer, you can mark out every place where you see clathrin and tell it, okay, look over now in the picture for AP2. See, is it colocalized over there? And what you see is that almost all the clathrin is in the same place as AP2. A small amount of clathrin is not localized with it. And okay, so if you do experiments, you realize there's always a little bit of noise, a little bit of slush. This is biology, that's the noise. You map out now where is the AP2. Almost all of the AP2 is in the same place as clathrin. A small amount of it isn't. And we say, okay, this is an experiment. There's always a certain amount of error. That's the noise. So this is exactly the kind of mistake that you want to avoid in your experiments. If you read almost every textbook, they say clathrin comes to the vesicle. It's part of the vesicle. In fact, if you look at the vesicles going in the textbooks, you see AP2 on the vesicles. So this is a classic example of what we in the lab refer to as the school bus problem. So most of you are from New York City, or at least the New York City environment. So I think you're familiar with these guys. You've seen them before? This actually is not a trivial question. It turns out that most of the members of my lab come from other countries. So I have people in my lab from India, France, Sweden, England. It turns out in none of these countries do they have school buses. Students go to school on public transportation. I think the one country I heard of, in certain parts of Australia, they do have some school buses, but they're white. So when postdocs come to my lab, people come to my lab to work from other countries, what really impresses them when they come to New York is not the tall buildings, because you've got tall buildings sort of in every country you go to. For Jyoti from India, from Stefan from Sweden, the tall buildings. It's not the people who come down the streets and borrow your wallet, because that happens in other countries also. What really impresses them when they come here are these things, because they don't exist in other countries. So let's say we assign them the job of figuring out, what does this thing do? So you give them a fancy American spy plane, you have them fly over and start taking pictures. And you'll get pictures that look like this. I think you've seen scenes like this before. So it turns out, being scientists, you can go through and you can start asking, okay, what can we learn about the function of these school buses from this? So you start doing an analysis and you ask, what do they localize with? Who are they found with? And if you do an analysis, you find that school buses co-localize with barbed wire and guard dogs. And in fact, statistically, there's an extremely strong coefficient of statistical significance, clearly saying this must be what is something related to the normal function of school buses. Now, if you take enough pictures of school buses over time, you'll occasionally get a picture of a school bus like this, moving down the street, picking up kids or dropping off kids. It's roughly 0.02% of the school buses. So clearly, it's not related to the normal function of school buses, the normal physiological function. It turns out it's a similar problem with the experiment that we just did with clathrin and AP2. So I mentioned there was a lot of clathrin on the surface. Most of those little spots are just sitting there. They're not doing anything. It was only a very small number that were moving into the cell. It was 0.01% in each frame. If we start looking at the various spots of clathrin, the ones that were still on the surface, they had clathrin and AP2. The ones that were moving had only clathrin. And for those who are close up, there's a measurement there for AP2. The error bar is there, but it's sort of hard to see. If you then look at the ones that are internalizing into the cell, they have clathrin, not AP2. This is a very small percentage of the population, but this is the population that we think is internalizing things. These ones that are disappearing, these are the ones that have the cargo. These are the ones that have brought together all the different components involved for internalization. The problem is, by looking at the average population, what we were doing is we were looking at the school buses in the parking lot. We were looking at the school buses co-localizing with the guard dogs and the barbed wire. Now, there are a number of ways you could get around this. For example, you could try synchronizing your observations, taking pictures at different time points during the day. And if you're lucky, you might happen to catch that right around 7 o'clock in the morning, a lot of the school buses were no longer in the parking lot. Or right around 3:30 or 4, they weren't there. Or right around 5, they were at the donut shops. But if you can synchronize, you might get information you wouldn't get from just doing these macroscopic observations. So often in biology, the important event is a transient one. It's localized in time, meaning it occurs quickly. It's localized to one small region. When you just look at the average properties, you miss it. Let me go to the next issue, the next advantage of studying individual events. Very often, it's hard to put a temporal order to events. I'll give you a few examples of that. So let's say that we're the first generation of humans who get to travel to another planet. And we see that it's inhabited, and let's say we want to learn something about the behavior of the people who are living there. So one way you can do it is, so let's say this is the planet we happen to come to. We could use our ray guns, zap the entire population, and then come in and start studying what were the people doing, what were the individuals doing. And what you might find is that some of them were consumers. We caught them in the process of taking in food of various forms. Some of them, we might decide, are hunters and gatherers. We find them in the process of, out on whatever errands. Others are maters, involved in various reproductive activities. That was a picture from the lunch downstairs for those of you in the faculty club. And others were thinkers, people who were involved deep in meditation. The problem with this, then, is that we're just sort of catching the entire population, and once we're averaging them, we think they are separate populations of people. Now, if we were really very astute when we came in with our ray gun and we killed everyone, we might notice that, in fact, the entire planet wasn't lit up in light all at the same time. So if we happen to remember where we were taking people from, we might have noticed that over in the areas that were lit, we had more of the consumers, people that were eating, or more of the hunters and gatherers. Or if we looked over in the dark regions, we may have noticed that there were more of the maters, or more of the deep thinkers. Now, if we did this kind of study, we might come to the conclusion then that since we mainly saw consumers and hunter-gatherers here, that light is inducing the birth of hunter-gatherers and eaters. Right? So if you were in the light areas, the light induced you to be that when you grew up. Whereas if you were in the dark area, dark was inducing the birth of maters and thinkers. The problem is, with this kind of analysis, we're missing out on the time course. We have no sense of one thing developing into another. Now, if we were very, very careful in what we had done, and let's say instead of killing everyone on the planet at once, we came in and just killed a few and took a measurement, and then killed a few more and took a measurement, we might find that actually the population is shifting over time as it moves along, that in fact, we're sort of synchronizing the population. The sunlight is in effect synchronizing. We'll see that as the sunlight moves, the population is shifting from one to another. So it's not as if you're statically just born to be a consumer or just born to be a hunter, but in fact, it changes over time. Now, perhaps the easiest way to address this, an alternative approach, would be to follow a single individual over time. And if you follow just one individual at a time, you get a real sense of a person progressing from one stage to the other. It's not as if these are 4 different populations. It's the same population that changes from one to another. It's that sense of time course that you miss when you average, which you can get from studying individuals. Now, the problem with studying individuals is that the signal you get very much depends upon who you follow. So, for example, if you happen to be following a miner, or let's say someone who did the night watchman job, you might find them getting up at night, which is very different from others going to sleep. So who you follow affects what you see. Or another way of viewing it, imagine you came to Earth and you ended up landing at 1600 Pennsylvania Avenue, also known as the White House. Unless you follow the main individual who lives there around for a day. You might get one particular sense of human behavior that might be very different than if, let's say, someone landed on your shoulder and followed you around for a day. Hopefully your behavior will be different, but. So let me show how this applies actually to research. How if we follow things, we can get a sense. So I mentioned this, AP2, this molecule, is found with clathrin on the surface, yet we don't see it internalizing. So how can we describe that? So there are a few possibilities. It could be that clathrin appears on the surface and just disappears all the time, taking things in, and in fact has nothing to do with AP2. All the static stuff just sitting there is like the school buses in the parking garage. They're just hanging around and not doing anything. A second possibility is that maybe, in fact, the clathrin and AP2 are working together. They get together doing something, and then clathrin splits off from the AP2 and then internalizes, leaving the AP2 behind. A third possibility is that maybe they do work together, but then the AP2 disappears first, and then clathrin does its business job of pulling things in to internalize. As we saw just from looking at the average population of clathrin or AP2 on the surface, or any proteins, we can't resolve between those. But if we follow individual spots on the surface, we can begin to separate them. And if we do that, what we see is that in some cases, if this is the fluorescence from AP2 and this is the fluorescence from clathrin, in fact, the clathrin and AP2 are together first, and what you see is the AP2 disappears first, and then the clathrin does its business job of pulling things in. Some cases it disappears far in advance, sometimes shortly before. In other cases, what we see is that the clathrin and the AP2 are together, and then part of the clathrin splits off and then internalizes, leaving the AP2 behind. These kinds of questions of the temporal order of things we couldn't get from just measuring the average population. We had to follow individual molecules on the surface. So for those of you who are particularly interested in internalization, which I think may encompass just myself, the important conclusion is that AP2 is associated with machinery and dissociates just before. It seems to be acting almost like a little chaperone, bringing cargo to the clathrin, which then allows it to come in. The important lesson for all of you, though, is that these kinds of experiments can only be done by using microscopy to follow the activity, sort of by imaging, visualizing what's happening going on. By doing that, we can now resolve between these different models of what's happening. Let me give you 2 other examples. How does cargo on the surface of a cell get sorted for this internalization? So the approach will be to follow individual molecules. And the example I'm going to give you is a molecule called epidermal growth factor receptor. And this molecule is of interest for a number of reasons, one of which being that increases of activity of this receptor on the surface of the cell has been tightly tied in with various forms of cancer, particularly liver, lung cancer. Not only is its activity increased in lung cancer, but various treatments that lower its activity decrease lung cancer. So there are right now quite a few clinical trials underway in which people are using antibodies against this receptor to lower its activity. So if we can understand the process of sorting and internalization, we could have a handle on a way of decreasing its activity on the surface of a cell by bringing it in. So our approach was to express this receptor fused to the jellyfish green fluorescent protein, GFP, and follow its movement. So if you look at the receptor on the surface of the cell, normally it's diffuse all over. However, if we then add in a small amount of the molecule that binds to it, so this is called epidermal growth factor receptor, so we add in a small amount of epidermal growth factor, and within 6 minutes we see all of these receptors, which were all over the surface, suddenly clump together into little spots on the surface. And if we look at clathrin and the EGF receptor at the same time, clathrin, we said, normally is in little spots. When we add in the epidermal growth factor, now the EGF receptor is in spots that, in fact, strongly co-localize with clathrin. And if you then look at one of these spots of epidermal growth factor, you can see it disappearing into the cell, and the clathrin, and only the clathrin that's in the same position, disappears at the same time and the same rate. So what's happening is you're getting the growth factor binds onto its receptor. The receptor is now joined together into spots that get internalized in. So the question is, let's say my right hand now is the receptor. When something binds to it, activating it, is it then causing clathrin on the inside of the cell to assemble there because you've now activated the receptor? And that's why you now see this together with its clathrin. Or once you activate this receptor, is it now able to bind to some pre-existing spot of clathrin? That becomes important for understanding the mechanism of what's going on and how we can regulate it. Is this thing inducing clathrin to get together and pull it in? Once you activate it, is it getting trapped by clathrin and then pulled in? The way to look at it is to label both at the same time, follow them, get to the point where they're both together, and then run the movie backwards and ask who was there first. Was it the EGF receptor that was there first, or was it the spot of clathrin on the surface? So what we've done now, we've added in the epidermal growth factor, wait till the receptors come to little spots, mark the spots, and then we ran the movie backwards to ask who was there first. And what we always observed is that if you ran the movie backwards, clathrin was there first. It was the EGF receptor that was getting recruited to it. This is a case where working out the temporal order of events was only possible by doing the imaging. So what is the role of AP2 in this process? And the way we tested it was simply by eliminating AP2. And one way you can do that is by putting in a piece of RNA that is the complement to the RNA that encodes AP2. In terms of if you put them together, they bind so tightly that now the protein machinery of the cell can no longer make new proteins from that RNA, so you can get rid of it. This just shows, let's say, if this is one of the proteins from AP2 that's normally being made, when we put in this RNA that silences it, you can see you start losing that protein being made. So what happens now to this EGF receptor if we get rid of AP2? Does it still get recruited to clathrin? So here's the EGF receptor diffuse on the surface. And could you just lower the lights in front here? This is a case where we added an EGF. You can now see it forms the little spots on the surface. If we now eliminate AP2, the EGF receptor is all over the surface. But now when we add an EGF, it's no longer getting recruited to clathrin. In other words, having this AP2 there is really like having a chaperone there that you need. So this receptor is normally diffuse all over the surface. After it binds onto its ligand, it's found in these little spots that co-localize with clathrin. And what happens is activated receptors being recruited to the clathrin. And this recruitment requires the activity of AP2. It's like the chaperone bringing it over. The most important conclusion for you is that working out this temporal order of events would not have been possible other than by following single events. I'll give you one last example of this point. These are events during apoptosis. So it turns out apoptosis is a form of cell death. So there are many ways by which cells can die. They can die from nasty poisons, they can die from lightsabers, zap them with a laser, or they can be induced to commit suicide. So it actually turns out that all cells, or pretty much all cells in multicellular organisms, are pre-programmed to commit suicide. Now, this may sound a little strange. Why should the cells of your body be pre-programmed to commit suicide? There are actually two major reasons why that's very advantageous. So in some cases, the body needs to get rid of certain cells during normal development. So for example, a tadpole, once it has its tail, when it's time to change into a frog, it triggers that programmed cell death in its tail, so the tail dies away. Or during development, we actually recapitulate some of the development of earlier animals. So during development, we initially have webbings between our fingers. The cells that form those webbings are then induced to die, so that way jewelry companies can sell us rings to wear. Now, there's another advantage to programmed cell death, and that is that very often cells in your body get hit by an invader or taken over by a parasite, a virus, a bacteria, or they just go aberrant. They go postal, and then they start growing abnormally, something we call cancer. And it turns out the cells of your body, the immune system, are able to recognize cells that have either been invaded or started growing aberrantly. And what the cells of the immune system can do is then trigger that cell to commit suicide through this process that's called apoptosis. So I'm going to tell you about some work we've been doing to try to identify the steps of events during apoptosis. This is the work of Patrick Bowler, who's a graduate student, and Alexa Matthias. So we know that a number of discrete changes occur in cells during apoptosis. You lose certain components of your mitochondria, absolutely essential for energy. You lose the voltage across the mitochondria, absolutely essential for energy. You activate certain enzymes within the cytosol that just chew up the cell, things called proteases. So what's the order of these events? So it turns out this is not an easy thing to figure out. So let's say we have a group of cells in a dish, and we do something noxious to them to induce this programmed cell death. What you might find is, let's say if you have 1 million cells, let's say if you want to look specifically at the case of release of one of the proteins in the mitochondria, it'll get released on the average at about the 5-hour time point, but there's a tremendous variability from cell to cell. So it'll be 5 hours plus or minus 4 hours. There are some cells that'll happen almost right away, some won't happen until 9 hours. Then you do the experiment again and you look at when do they lose the voltage across the mitochondria, something you need for generating ATP. Again, it occurs on the average about 5 hours, but there's a huge spread. When do you activate these enzymes that chew up things in the cytosol? It turns out there's such a spread in when these things occur that you cannot work out a temporal order for which they occur. So the approach they took was to follow all of these events at the same time in the same cell. So they assayed the membrane potential by using fluorescent dyes, the release of components from the mitochondria with other fluorescent proteins, activation of the proteases, I'll tell you about in a moment. So let's say you just look at the mitochondria. Here's a cell where you're looking at one particular component in the mitochondria called cytochrome C, and you can see now the cytochrome C being lost from the mitochondria. You can even see when it's being lost from individual mitochondria as time goes on. Well, let's say at the same time we monitor the loss of membrane potential in the mitochondria. So you're going to be seeing cytochrome C in green and the membrane potentials in red. And you see that first they lose the cytochrome C, which is in green, and only afterwards do they lose the red. Or if you look here in the order, they always lose the cytochrome C first, then they lose their membrane potential. Now, the difference between these things is on the order of about 100 seconds. One happens and the other one's 100 seconds later. If you're looking at this in cells where from cell to cell there's a variability of plus or minus 4 hours, you can never work out the temporal order of these events. It turns out this is when they lose cytochrome C, this is when they lose another protein, SMEC. Or let's say we ask, when do they activate their proteases? The way Patrick assayed this was to attach a fluorescent protein to the outer membrane of a cell via a very particular protein that we knew would get chewed up very, very well by this enzyme. So what will happen is if this enzyme gets activated that chews things up, this fluorescence, which is around the outside of the cell, should now move into the middle of the cell. So you can see in this cell, you're getting activation of the protease. In fact, you can set up different reporters for different proteases, and you can begin to ask what happens first. So if you look here, cytochrome C is lost first, then you activate the protease. There's a difference of just a few minutes in a single cell, something you couldn't get from taking average populations where there's a wide diversity of plus and minus a spread of over 8 hours. But looking at the single cell level, you can resolve things that reproducibly have an order of events. So this allows us to follow the biochemical steps from looking at a single cell level. So what I'll leave you with last is, in terms of why is imaging having an impact, is that from macroscopic measurements, often there are many different microscopic explanations that can account for the events. And to illustrate this, I'm going to shift slightly. Instead of looking at membrane transport, look at another event that occurs on the surface of cells, and that is assembly of viruses. And this is work that actually has been done in collaboration with Paul Benoist, who's here at Rockefeller University, who's a virologist who studies HIV, and Nolan Juvenet. And the question we want to ask is, how does HIV exit cells? Where does it assemble? And there are two different models, one of which says that HIV buds right off the plasma membrane of cells, in which case the prediction would be is that viruses should appear here at the surface of a cell one at a time. The second model is that HIV assembles inside of the cell on these internal organelles, and then once they're assembled there, these organelles come to the surface, and through the same kinds of secretion I told you before, they fuse and release their contents. So we already know how to look at secretion. We've done that before. So the question is, do we see the viruses appear one at a time, or do we see a large number of them together, coming together and coming up to the surface? So the approach we took was actually to stick into the middle of the genome encoding HIV a green fluorescent protein, and then monitor the surface of the cells using the same kind of techniques I told you about before and ask, do they appear one at a time or many at once? So can you kill the lights in the front, please? So this is looking now at the surface of a cell, and what you should start seeing is single discrete spots appearing, almost like stars appearing at night. We only saw single individual spots appearing at a time. We never saw a whole group of spots being delivered to the surface at once, almost as if which is what you would expect if many of these viruses were assembling inside of the cell and then being released. I think so far we've seen somewhere in excess of 20,000 discrete spots, and they've only appeared one at a time. Here's a close-up of one of them so you can see a little better. So if you look here, you can see here's one, and then you'll see two more of these spots appearing. The problem is, how do we know we're actually seeing a virus assembling? And it turns out that, in fact, no one had ever seen any virus of any sort before. So how do you know you're really seeing real viral assembly at this point? So we decided we would use a few criteria. And this is something you have to be very careful of, because when you see something that no one's ever seen before, you want to really be sure you're looking at what you hope to be looking at. So one should be that if we're really observing assembly, they should be packing up so close together that all the molecules should be right up against each other. Two, if they're really observing assembly, then each one of those bright spots should represent a fixed number of viral proteins that are coming together to form the virus, a fixed number of molecules. And that once they've assembled, that should be it. There should be no other molecules coming in or out. And then third, if this was really a virus that was coming together, assembling and budding off, it should be separated off from the cell so that things on the inside of this virus should no longer be able to exchange with anything inside of the cell, no matter how small the molecule is. So how can we judge how close they're getting to each other? It turns out that if we use a number of different fluorescent proteins have this very weird phenomena that when you pack them really, really close to each other, one fluorescent protein can excite the next fluorescent protein. Now, when I mean really close, they've got to be on the order of a single molecule away from each other. It turns out this effect falls off by the distance to the 6th power. Right, so if you move just twice far away, it drops off to just 1/64th of the effect. You go 3 times as far away. I'll open my Excel spreadsheet and tell you afterwards. The critical thing is that they've got to be on the order of a few nanometers apart. So if we go in and let's say we have a green fluorescent protein and a cherry fluorescent protein at the same time, what will happen is if we then excite the green, we should be able to have the green in turn exciting the red protein, which is what we observed. So in fact, we were able to tell these molecules were in fact packing very, very close to each other. Then we were really recruiting a fixed number of molecules here, or was it more sort of like this kind of mixture? Was it a sort of aggregation of things together? So the test we did to ask were we really getting a fixed number of molecules was to come in and bleach all of the fluorophores over here by giving a very sharp, short laser pulse. If we did that, if nothing else was being recruited to it, the fluorescence should be dead. Whereas if it was more like an aggregate here, if you kill the fluorescence, new ones will be coming in and the fluorescence should recover. So if you look over here, here's a particular spot. It's a little weak. It's getting stronger. After we bleached it, you see now that it's recovering back up. And actually, here's the quantification of that one. So it was growing. We bleached it. It came back. Here's one of those bright spots that had already reached its steady state. Once we bleached it, it no longer came back. It was already at the point where it had separated off from the cell. It was no longer recruiting in new molecules that were fluorescent. So now the question is, have it really fissioned off from the cell? Has it really separated off from the cell surface? So what we want to know is, has this virus gotten to the point where it can no longer exchange any molecules from inside the virus to the cell? So what did I ask is, what is the smallest molecule we can ask that can it get from inside of the virus to a cell? So what's an example of a small molecule? Water, smaller. Hydrogen. Hydrogen was the smallest one I was able to come up with for the time. So the way we tested it was to try to increase the number of hydrogen ions in the cell and see would they get inside the virus or lower the number of hydrogen ions in the cell. And the way I did it was to take advantage of this reaction right here. So it turns out that whenever the carbon dioxide raises in your body due to metabolism, you have carbon dioxide gets together with water, and thanks to an enzyme called carbonic anhydrase, you produce protons and carbonate. This is what helps us, it's actually the reaction going the other way. This is what helps you get rid of all the acid you're producing when you're exercising and running. And this reaction goes very, very quickly in both directions, thanks to this one particular enzyme, carbonic anhydrase, that works incredibly quickly. So what our approach was the following. In this molecule here, in this potential virus here, we had proteins that had a fluorescent protein on them. We changed it for another fluorescent protein that was very sensitive to pH. So if things became more acid, its pH would change. If it became less acid, the pH would change. And we could monitor whether or not protons can get in there. So the idea was now to raise or lower the protons here and see could they be detected inside of here. So the way we changed the number of protons inside of the cell was simply by raising or lowering the pH. So if you raising or lowering the CO2. So if you lower the CO2, you become more alkaline. You raise the CO2, you become more acidic. If you just switch back and forth like this, the cell responds within seconds. So this is now looking at some of these spots of HIV on the surface. We want to know have they really separated off. And right here at this little green line is where we gave a very brief pulse of CO2 to make them slightly more acidic. So what we're going to look at first is just some of the protein from the HIV that was still back in the cytosol that had not yet assembled. And when you looked at that, when you gave a pulse of CO2, it shifted very rapidly. But then after the CO2 is over, it recovers very quickly. Now we're going to look at some viruses that had already been budded off from the cell. These were viruses we had collected out in the supernatant. They were already moving onward to affect other cells. When we looked at those, when we gave a pulse of CO2, they didn't change as quickly. And they definitely didn't recover as quickly. These viruses that had separated off from the cell were not as sensitive to the CO2 because they don't have carbonic anhydrase. So now the critical question, for these spots that we saw assembling on the cell, were they really representing viruses assembling or not? So we looked at their response to CO2. This one here in red that was still increasing in fluorescence, it was very sensitive, meaning this one was still exposed to the inside of the cell. Protons can get in and out very quickly. But this one here in blue that had already reached its maximum fluorescence, it didn't get quenched as far, and it didn't recover as quickly. This one had already budded off from the cell. It can no longer exchange even something as small as a proton with the rest of the cell. So if you look at some of these things very early in assembly, you see that this is the degree to which you can quench them down with the carbon dioxide. Once they bud off, they're much less sensitive. As assembly goes on, they shift over to this resistant form. They've budded off, they've separated from the cell. So from looking at these single events, we can now work out the steps in assembly of the virus. Actually, now we can distinguish when does the mRNA come in? When do you activate other proteins in the process? So the question is, if looking at single events is such a powerful approach in biology, why doesn't everybody do it? And there are a number of reasons why, in fact, not everyone embraces this technique. So one, if you're going to label molecules, you have to worry, is your labeled protein functional? It turns out very often they're not. The second one is, how do you know you're really observing the single event you want to be observing? That's not trivial, but it's often a source of mistakes, and I'm happy to discuss it afterwards. How do you know if the signal you're reporting by your assay is really representing the event you want to study? When you're seeing something new for the first time, you never really know if you're observing what you really want to be observing or if you're observing some artifact. But the final point is perhaps the most difficult. How do you know when a single rare event you're seeing, a single anecdotal event, is really important? And this is actually a very difficult problem. So people have argued that by having increased sensitivity is actually going to be disadvantageous for a lot of purposes. So for example, in medical diagnoses, it's claimed that if your imaging, let's say your microscopes, your ways of visualizing things in the cell become so sensitive, then we're going to start detecting pathologies in everyone, and that's going to be a problem. Or they would argue that if your imaging is sensitive enough, your ability to see and detect things with your microscopes, then we can start characterizing the individual molecules, in this case the individual school buses, and determine which ones are significant and avoid unnecessary treatment, or which ones are significant. But this is actually a rather difficult problem. How do you know when a rare observation is something very significant? It's something that, well, let's take it this way. Let's define a miracle as a 1 in a million event, okay? Something that's extremely rare. So it turns out if you walk down the streets of Manhattan, you tend to see something new pretty much every second walking down the street, which means roughly you see 60 new things a minute, or 3,600 new things an hour, or you'll see roughly 57,000 new things on a typical day, which means that if you, a 16-hour day, which means that roughly once every 17 days of walking down the streets of Manhattan, you're going to see a 1 in a million event, a miracle. Now, if you consider that on a typical day, there's roughly 15 million people walking around Manhattan, that means you have, on a typical day, 15 miracles being observed a second in Manhattan. Now, one approach could say, okay, that's life in New York City. But as scientists, we don't usually invoke miracles. We invoke statistics. We're basically saying, if you throw the dice enough times, you're eventually going to get sixes in a row. Or if you observe enough events, eventually you're going to see some weird events. Most of you from New York are sort of familiar with this. I've tried to raise this point with people outside New York City where life is at a slower pace. They're not accustomed to seeing new things as often. So but I've discovered that even out in Iowa and Utah, they've heard of eBay. So the example that tends to work for them, how many people here are familiar with the grilled cheese sandwich that's sold on eBay for $25,000? Yes. Turns out a guy who worked down in a diner in Florida was flipping grilled cheeses, and it turns out after flipping grilled cheeses day after day, came up with a grilled cheese that had the face of Madonna, which he sold on eBay for, I think it was $25,000. How much was it? If you think this is an urban myth, you can actually go to Amazon, and there's a book for sale on Amazon about some of the top-selling items on eBay, and it's called, I think, the Grilled Cheese Mary or something. The point being is if you flip enough grilled cheese sandwiches, you observe enough events, eventually you can see almost anything. So the problem becomes, if you're seeing a rare event, like that thing of Clathrin going in, I said it was a 1 in 10,000 event, how do you know if it's something of significance? And that's a non-trivial problem. So the best test we can come up with is that if we add up each of the individual events, can account for the behavior we're interested in. So in the case of Clathrin, we know the whole surface gets internalized once every 10 minutes. Given that it's getting turned over once every 10 minutes, there should be only a certain rate, if you're seeing things once every 10 milliseconds, that it should be happening. So in fact, if we were observing it at a much higher rate, there'd be a problem. In other words, each of the individual events has to add up to the behavior of the population. So why is imaging having such an impact? At the very beginning, I gave you 4 reasons. The last one being is that imaging allows us to examine a multitude of sizes simultaneously, from single molecules to whole organisms. So for example, the question of how is ATP generated in our mitochondria? It turns out we can watch the individual proteins in the cell as they generate ATP right now. Whoops. There we go. So what you're seeing on the left, this is an individual protein machine that is generating ATP. And the way it works is that as it drives protons across, it actually spins around, rotates around. On the right is an actual model of it in the process. But the point is we can watch single molecules as they work in the living cell as a way of addressing questions of how do they work. I already showed you examples of how you can see groups of molecules working. I gave you the example of secretion or the assembly of viruses. You've already seen as you move up, you can see blood flow through the heart, through the retina. We can now also image larger scales. We can image activity in the brain or individual branches of nerve cells or activities in regions of the brain. And we can also watch single tumor cells as they metastasize through the body. So why would you want to image single tumor cells? Let me give you one example. We have a collection of tumor cells that range in their metastatic potential, from some that are non-metastatic, these happen to be melanoma cells, to those that are very, very highly metastatic. So the question is, what's the difference between them? What makes one cell much more metastatic than another? Is it the ability of them to bind to the inside of the blood vessels? So maybe they go through your blood system, and only the highly metastatic ones can bind inside your blood vessels. Or maybe they can all bind to the blood vessels equally, but only the highly metastatic ones can cross blood vessels. Or maybe they can all cross equally, but only the highly metastatic ones can stimulate the local release of different enzymes that allow them to eat their way through. Or maybe they can all invade equally well, but there's difference of how well they can survive in response to attacks by the immune system. So if I just took, let's say, 3 different kinds of tumor cells, the highly metastatic, the slightly and non-metastatic, and let's say I injected 30 of the highly metastatic ones into 30 of you, 30 of the slightly metastatic to 30 others. We came back in a year. What we discovered is that a group of you have tumors and a group of you don't, hopefully. But we wouldn't resolve between those different mechanisms. We would also have a court suit. We would also, anyway. It wouldn't help us to resolve what is responsible for the difference between them. So our approach was to load individual tumor cells with fluorophores. In this case, we used something called quantum dots. So we had them colored so we can actually distinguish between the highly metastatic and the non-metastatic cells in the same mouse at the same time. So you could, using microscopes now, you can actually image into the tissue through the skin and in the lung. And here you can see the different lung cells. This blue here is marking the inside of the blood vessels. And they begin to ask questions of that sort. So it turns out that if you just look 3 hours after injecting in the cells, the number of highly metastatic cells is the same as the number of slightly metastatic cells, which is the same as the number of non-metastatic. In other words, they all bound onto the blood vessels equally well. They all crossed over equally well. They all invaded the tissue equally well. So for me, this was rather disturbing. I'd always learned that metastasis has to do with how aggressive a cell is at invading your body. But they were all invading equally well. Well, what happened if we looked a little bit later? Let's say we looked instead of 3 hours, 3 days later. By 3 days later, what we saw now is that we had many more of the highly metastatic cells and fewer of the slightly or non-metastatic cells. So what was the difference? Was it that these cells were dying off, or were these cells actually growing? So the way we tried to address the question was to load these cells with a fixed number of fluorophores. That's why I use this molecule called quantum dots, where every time a cell divides, it would dilute out the number of these fluorescent particles. And so as they divide down, they should become less fluorescent. And what we found is if we looked at just the slightly metastatic cells, their fluorescence didn't really decrease by the time you got to 3 days. But for the highly metastatic ones, it did decrease. They were very actively dividing. So the difference in how metastatic they were was not how effectively they invaded. It's to what extent, once they invaded, they observed the local societal rules and didn't grow, or they ignored them and they started growing. This is something you could not evaluate from looking at a macroscopic level of how many tumors you get in the body. You have to look at the level of the single cell. So the conclusions from this, and I'm going to emphasize over again that these conclusions are always temporary, is that metastasis may not be determined by how aggressively it binds or crosses. It may reflect whether or not a cell grows or not once it's invaded to an area. And these questions have not been accessible from looking at tumor growth. They required sort of advanced microscopes to be able to image in and follow single cells. So within the near future, there are a number of possibilities that we can do. We can use various forms of microscopes to look in more than one modality. Instead of just looking at optical light, we can use something called functional magnetic resonance imaging or positron emission tomography simultaneously. Right now, we're using imaging solely to evaluate various therapeutic treatments, but we could also use it for diagnostic purposes. For example, we know that different schizophrenics respond differently to different drugs, not from personal experience. We know that certain drugs work with some schizophrenics and not with others. Perhaps by doing imaging inside of the brain, we can actually in advance determine which ones will be more effective for some patients than others. Actually use it to diagnose the treatments. And actually use brain imaging, various forms of microscopies, not just optical imaging, to see what's the preferred treatment. So why image multiple sizes? So many diseases are the result of something that's defective at the molecular level. You've got a mutation in a particular protein, and it's not functioning right. But these are observed as diseases on the systemic level. It's the patient who comes in that's ill. What we'd really like to be able to do is be able to follow this pathological process at the level of the single molecules. Let's say if you're interested in cystic fibrosis, see what's wrong with that particular molecule, or in muscular dystrophy, what's wrong with dystrophin or the other molecules. At the level of the organelles that they're affecting, at the level of single cells, at the level of cellular systems, whole organs, and then at the level of the whole patient. And what imaging is allowing us to do is study these things from the single molecule all the way up to the whole individual in a living individual. And I think what you're going to start seeing over the coming years as you graduate high school, go into college, is you're going to start seeing imaging as having more and more of an impact on our society on all different levels. What's a good way to illustrate it? So for example, last year, my daughter was studying Greek civilization, and she's hiding down in the front row. And the way they studied Greek civilization was by studying its writers and philosophers at the time. So what do? They read the work of Homer. So if you want to see the kind of impact that imaging is having on society today, what you have to do is sort of read the work of the philosophers and poets of our age. Or effectively, what you have to be doing is sort of reading the work of the Homers of our age. Is your volume off? Hello up there. So in the beginning, I told you there are going to be a number of different strands to the talk. First was the issue of an approach to science, and that is specifically trying to look at individual single events that we covered. What I hope I've shown you is that from studying single events, the macroscopic population is revealing an average that may not be meaningful, particularly if you have two separate populations. Or the average may represent the dominant population, but the stuff we're interested in is like one of the rare transient events. But also from looking at individuals, we can now assign a temporal order to events. And we can start looking at microscopic mechanisms. So for example, on a macroscopic level, when we're looking at HIV going out from a cell, we couldn't tell whether it's just being made one by one on the surface, or whether it was assembling inside of the cell and going out in mass. From looking at single viruses, we could distinguish between those two mechanisms, which is important if you want to start designing treatments for interfering with HIV assembly. Why imaging? What I hope I've shown you is that imaging is having an impact because it allows us to study living systems. It allows us to stimulate or activate different pathways. It gives us information about the variations, the diversity of a system, allowing us to study individual events, and allows us to study a multitude of sizes simultaneously, from whole cells to whole organisms. And what I gave you were a few different model systems, how cells secrete, grow, respond to signals in the environment, how viruses are made in cells. And the most important conclusions for you to take home are that there are things you can learn from studying single events you cannot get from studying populations, and that imaging is allowing us to address questions that even a few years ago were considered unaddressable, inaccessible in science. And perhaps most importantly, this is the most exciting time ever in history to be studying science. Now, the images that I showed you are just a small sample of the kinds of things that are being done today. If you want, there's a reference list that's available in the back. So all the images that I was showing before the talk began, in the morning of pond life, and later in the afternoon, most of them of mammalian cells, are available at this wonderful site that's listed on the reference list run by Florida State University, which also has a great tutorial on microscopy, all the different modes of microscopy and things you can do with it. And they also have all those videos I showed you. For further information on the photoactivation of fluorophores, here's an example of a paper from Tom Muir, but this is also listed on the list outside. The blood flow through the heart was from the work of Scott Fraser and colleagues. Blood flow to the back of the retina, Marty Freelander. For more information on the use of GFP as a carrier, if you go to the site noble.se, it's the Nobel Awards website, you can actually listen to the Nobel lectures by the 3 laureates who just got the prize for GFP. And they're incredibly articulate. They've got great images. The activity of the proton ATPase was from the work of Yoshita and colleagues, and the modeling was done by George Oster. All the other work is the work from people in my laboratory, which means I should start off by thanking a few people. First, the National Science Foundation, who's been supporting my work for 39 years. First, as a high school student, both taking stuff at Columbia University, at Cornell, later on as an undergraduate. And even today, the NSF is providing funding for almost all of the innovative and risky work in my lab. The NSF has an extremely small budget. I think it's most probably less than one of these experimental planes that's being used by the military, which is sad but true. But they have a tremendous impact. I also want to thank the American Cancer Society. They've provided a lot of the seed money for the cancer stuff that we're doing. And they not only fund very important research, they're a very, very valuable resource for patients who have cancer. And if you're looking for a group to support, I highly recommend them. And then the National Institutes of Health, who provides the vast bulk of support that drives research at this company. I also want to thank my parents, who've always supported my interest in science. Some amazing teachers that I've had. I'm in high school, despite one bad experience, actually I had two very good experiences. I did finally get a rather depressing grade in that 9th grade class, but the chairman of the science department, George Kapp, actually read the answers that I had written instead of marking down A, B, C, and D. He fortunately overrode the teacher and changed my grade. I also had a biology teacher who was very helpful. In college, I had 2 great advisors, and actually, one thing I should mention, that science is still run very much like an apprenticeship. It's sort of like training in the arts, where you work with a particular person and become trained in the work. And I've actually had 3 great mentors. Robert Richardson, which is the first lab I worked in at Cornell, Rodolfo Linas, Gunter Blobel here at Rockefeller. I also want to thank the leadership here at Rockefeller, particularly 2 presidents, the current one, Paul Nurse, and Torsten Wiesel, who was here throughout the 90s, both of whom have put a tremendous emphasis on supporting research that is really pioneering. Their importance is not that you are the person getting the most grant money, the person being invited to most symposium, but they want you to take risks, which means also things failing. And they've really been, both them and the Board of Trustees have been wonderful in that extent. I also want to thank all the members of my lab, both past and present. Science is very much a teamwork of people working together, and they've been great. And finally, I want to thank my family, who and one last final note. I also want to thank Bonnie Kaiser, who introduced me at the beginning. Bonnie runs a magnificent science outreach program where not only high school students, but high school teachers come in. Francesco, you still here? Yeah. Francesca worked in my lab during the summer, as well as numerous other teachers. And she also reminded me at the very beginning of a quote, which should be the mantra for this talk, from Yogi Berra, saying, "You can observe a lot just by watching." Thank you very much. So you have a choice. If there's any really burning questions, why don't we take a few questions up at the microphones now, and then after like 5-10 minutes, the bulk of you can go, and anyone who has any remaining questions, you can come up.
What seemed to be missing from everything in your work seemed to be electron microscopy and scanning electron microscopy. I know they cannot be used basically with any specimen that's wet, with biological specimens, but I thought with the type of work that was doing, there should be some impact or some use of it. And I just was surprised at its absence.
So it turns out there are many, many different ways by which you can image things nowadays. You can use light to image things. You can use electrons. So the advantage of electrons, I mentioned that our sensitivity is due to the wavelength of light. An electron has a much, much shorter wavelength, allowing you to see finer detail. The only potential problem there is that you have to sort of fix the cell. You have to pickle it. So you can't look at things dynamically. It's a very powerful technique. It's helped provide a lot of insights. And there are advantages to using a multitude of techniques for every problem you go after. So not only using light microscopy, but also using genetics, also using biochemistry, electron microscopy. Beyond that, we're actually looking also at other imaging modalities beyond just the optical. I mentioned fMRI and PET scanning. No one technique is sufficient for addressing anything in science. So that's a very, very important point that was just raised. Another question?
What would your answer be to people, medical people who are saying that when you do this imaging, you get too many false positives and it's too expensive to do because then you have to go to the next step. And so a lot of new techniques of diagnosis they don't want to do because of money at the next step.
Mm-hmm. So one of the problems with these techniques is that as we increase the sensitivity, you have a trade-off. You have the potential of getting more things that say that they're positive when they're not really. And so you often want to do this kind of imaging to avoid unnecessary surgery, but you don't want the surgery to say, oh, you've got something there when you don't, and then have a surgery that's not really needed, which besides being expensive, it can be a real pain in the, well, it depends where the surgery is. I think what's required then are better probes for what you want to look at. So right now, for example, if you want to look for a tumor, the main approach they have is to give in radioactive sugars, and the cells that are most active metabolically tend to be the tumor cells. The problem is, if you have immune cells that are fighting something like a cold, they will also light up. Brain cells light up beautifully with this technique. The problem is the technique is not specific enough, and what we need are new ways of getting specific, and there are a number of possibilities for that are just being tested now. But that's what's needed. But my feeling is that if we start, I think what's needed is more sensitive and specific techniques, because often if you catch things earlier, the cost is a lot less than if you catch it later on. And I don't mean only cost in terms of the medical costs. There are also costs that when you catch something later on, that person then is out of circulation for an extended period of time. They're not contributing. All of the members of their family are then out of circulation. By catching things earlier on, there's less of a cost, not only in terms of initial dollars, but long-term cost to the society. I think what we also need, which gets beyond that, is issues of more prevention, more issues of personalized medicine that are targeted to those things. So it is a problem now, but I think it is a surmountable problem. Any other burning questions? So the question is, how do you know that actually attaching this fluorescent protein on isn't affecting your protein? So it turns out that is actually a very serious problem. So this gets back to the question, are the labeled molecules function? Often they aren't. So it turns out that the cystic fibrosis protein that I referred to before, it turns out that if you put at one end of it any kind of tag, not only a green fluorescent protein, even a very small tag, you totally change where the protein goes in a cell. If you put the same tag at the other end of the protein, it goes to the right location, but there are very subtle differences in its behavior. So the problem is that it's very important to demonstrate that when you've labeled a protein that it is functional. So there are a few ways of doing that. One way you can do is to knock down or eliminate the protein that's normally there in the cell. I mentioned one technique of using an RNA that will hybridize, knocking it down. There are poisons you can use. So eliminate the protein that's normally there, in which case the cell will be sick, it'll be not functioning. Now come in with your protein fused to your green fluorescent protein and ask, can it restore the lost function? Sometimes it does, often it doesn't. But that actually is one of the limitations in the technique. You've got to be sure the protein you're studying is not altered by your labeling. Very good point. Any other burning questions? The question that was asked is, why is it normally at night when they park school buses, are they protected by barbed wire? I think the corollary to that question is, what I don't understand is, why is it when the day is over and school buses are parked for the night, why any kids would ever want to go back to a school bus? But that's more philosophical. So how about, if anyone has any questions they want to ask on their own, you can come up. To everyone else, thank you very much for your attention, and have a great vacation.